which Vithanone showed, Bacoside also showed DNMT inhibition and HDAC2, HDAC5 inhibition and CBP upregulation. So could be that these molecules are further upstream acting as an epigenetic modifier. So this was all about Vithanone and Bacoside. So in parallel, we are also doing a cell-based screening of phytochemicals, and that includes these vithanolides and other compounds as well. And I'm not going detail into this study because I think next Dr. Renu Vadva will speak about honeybee propolis and stress exposure. So this is our recently published paper on CAPE. So what we did a high throughput screening of phytochemicals. Particularly, we targeted a phenomenon called neurodifferentiation. And we found that CAPE has an extensive neurodifferentiation property, not only in the primary cultures, but also in the neuroblastoma, glioma cell lines. And in vivo, its neurodifferentiating property was replicated in amelioration of cognitive and physiological deficits. So now my highlights of my studies. So I've shown you the findings from three major natural compounds which we're interested in: Withania somnifera leaf eye extract, it's bioactive Vithanone; Pacopa monnieri, and New Zealand honeybee propolis, it's bioactive CAPE. So I'm going to just summarize what are the overlapping and discrete properties of these three herbs. Is that we found that all the three compounds, they have a recovery of memory deficits potential, memory enhancement, and neurodifferentiation potential. Whereas eye extract and Bacopa also showed epigenetic modulation, neural regeneration, and neuritogenesis and elongation. And CAPE has an extensive neurodifferentiation property. So my future direction that I'll like to assess the combinatorial formulations of these compounds and their nanoconjugates in particular. We will do a detailed molecular analysis and at epigenetic level. And we'll move this scopolamine model further, we'll expand this model into transgenic animals and human iPSCs. So that's all. I would like to acknowledge the funding agencies, particularly DST Inspire Grant, Dialab and Grant, DST and DBT. So this work is a team work, which was, and so many people are associated. I have just tried to enlist few of them. So Professor N.K. Thakur and all the LOVE members, Professor Renu Vadva, Sunil Kaul from AIST, Dr. Vinay Khanna, IITR, Dr. D.K. Lahiri, and Dr. Veena Kaul from IIT Delhi. So thank you.
Thank you, Arpita. So the topic is open for questions. We can have one or two questions, please. Okay, so if there are no questions, we move on to the next speaker. Over to Dr. Simran, please.
Yes, sir. Thank you, sir. So may I invite Dr. Renu Vadva, Biomedical Research Institute leader, DBT AIST, Japan. So her specific research interest area is aging and cancer. And she's been awarded with great recognition. She's an editorial board of several scientific journals, including Journal of Gerontology, Biological Sciences, Mechanism of Aging and Development. She's a fellow of Geriatric Society of India, IAN, Biotech Research Society, and foreign fellow of National Academy of Sciences. Ma'am, may I please share the slides, ma'am? Thank you.
Yes, I'm trying to share. Do you see my slides?
Yeah,
yes, ma'am, they're visible.
Slideshow.
Okay.
Slideshow.
Yeah, just a minute. Yeah, my slides are there?
No, slideshow.
Yes, ma'am, but they are in presentation mode.
Okay. Just a minute. How's that?
Yes.
Yes, ma'am.
Okay.
Perfect.
Right. Hello, everyone. I'm happy to be with you all, although from a distance. I really hope the next meeting in Calcutta, probably, we will meet again. So today, with the shortage of time, I wouldn't go into too much of the talking in the beginning, and I'm going to present to you our work on honeybee propolis this time. A little bit unusual. For the last years, I've been presenting ashwagandha. Okay, so I'm sure you all know what honeybee propolis is. It's honey, everybody knows. Everybody knows about honey, and we love eating honey. Honeybee propolis is another sticky substance that honeybees make with their saliva and the botanical material they live on. And this is called propolis, or sometimes it's also honeybee wax. So it's known as a very popular health supplement Honeybee propolis is branded and labeled based on the region it comes from. New Zealand propolis is very, very popular in Australia, New Zealand, and Brazilian propolis is also very, very popular. So they differ in terms of their active component. New Zealand propolis has a cape, caffeic acid, phenethyl ester as a major component, and Brazilian propolis has artiplin C, and it has a lot of other, bioactives listed on my slide here as flavonoids, all those phenolic acid, and a number of papers are already there describing antioxidant and other activities listed on my slide. Now, I'm going to share with you our experience and the results and the data, how this honeybee propolis has walked into our lab and what we searched and what we know about it. So first of all, as a mainstream work of my lab on cancer, it came as an anti-cancer activity, and PhD student Nupur Nigam struggled with it for a while, and she got the results like it has anti-cancer activity for breast cancer cells and their metastatic versions, and it induces apoptosis and G1 arrest. And when I said she struggled, what she was finding was that when she's treating the cells for the short term, they were having cytotoxicity. But when she kept the same cells for 96 hours or longer, the cells were coming back, which was an indication that the active cytotoxic components of this propolis extract were not really stable. They were getting off, and that's why the cells were reviving back in the cell culture. So we kind of struggled with it for a while, maybe half a year or a year, and then we solved this issue in collaboration with a company called Cyclochem, which specializes in cyclodextrins. What we have come up with is encapsulate CAPE with gamma cyclodextrin, so we protect it from its degradation or hydroxylation, or by degradation by esterases, which are there in the culture medium or in the body. So this is one of the data showing that the CAPE gamma CD is much stable in terms of temperature, in terms of other digestions, reactions in oxidation reactions. So we have generated this version of CAPE, and then Nupur tested it in the cell culture medium. And as you see here, as compared to CAPE gamma CD activity was cytotoxic activity was much more stable, and it was reproducible in number of cell lines. Not only in cell culture, but also in vivo experiments, we got activity much better. In here, you can see that the CAPE gamma CD green plots are better activity, better anti-tumor activity as compared to CAPE as an individual component. So we worked through a couple of years on the mechanisms of cytotoxicity or anti-cancer activity of CAPE, and I wouldn't go into details. And just to summarize here, that there are a number of mechanisms: induction of DNA damage, dissociation of Morlin p53 complexes. So all this is published data, and this is the anti-cancer activity chapter of CAPE in my lab. So as I usually present, there are a number of projects going on in the lab, and one of them was hypoxia modulating factor, because we already know very, very clearly it is established that hypoxia has a major role to play in the brain diseases, age-related pathologies, and on the other hand, aggressive cancers. So we've been interested in hypoxia modulating factors. Another PhD student in the lab, Priyanshu Bhargava, he established a cell-based system where he stably transfected the cells with HIF-driven promoter, which is luciferase. And I wouldn't go into details what HIF is. It's a main hypoxia modulating or regulating transcription factor. So HIF-driven luciferase was used as an library screening system. And what he found was, out of nearly about 80 different natural and synthetic compounds run for the library, one of them, like out of the 4 here, which are inducing hypoxia or activating hypoxia signaling, is CAPE. So that's another door the CAPE came into my lab. So Priyanshu also did experiments to validate, yes, with CAPE-treated cells, you actually see more of luciferase, more of endogenous HIF1 alpha, showing that CAPE is a reliable inducer of hypoxia signaling. Then he also, if it is inducing hypoxia, our obvious question was whether it has something to do with protein aggregation, deaggregation phenomena. And Priyanshu again established heat shock driven protein aggregation or MITS folding, and then the metal stress driven sodium arsenite he uses, metal stress induced again aggregation. And in both assays, you see that the CAPE treated cells have nice luciferase protein expression when it was gone, when the cells were heated or heat shocked. Similarly, here down you see he's using GFP as a model system, causes its aggregation. You see in this panel, aggregation induced by sodium arsenate, and when the cells are treated with CAPE, we see deaggregation. So that was nice, and that told us that the CAPE has protein deaggregation activity. And you can imagine how important it is with the age-related brain dysfunction and pathologies which are driven by protein aggregations or the molecular garbage. Now, the third door through which CAPE walked into my lab was, as Arpita already explained, we've been interested in neurodifferentiation modulating factors. And she, when visited us, she undertook a screening, actually looked at the compounds which could induce neuronal differentiation in terms of very different criteria. And Rajkumar Kalra, staff with me here, he also contributing this in this project. And these beautiful data by Raj shows, yes, CAPE has a neurodifferentiation capability and as good as retinoic acid, which is an established neurodifferentiation agent. Now, we took this analysis in different cell lines, different molecular markers, all collectively showed that CAPE is a nice neurodifferentiation molecule, not only in one cell line, but also the others like GOTO cell line, PC12, and others they tested. Now, of course, there was a concern whether this phenomena is happening only in cell culture or we see in the animal models too. So in collaboration with Manipal University, Guru Prasad and the group, we took this assay. They are using Alzheimer's model Drosophila. And what I'll like to show you here is just look at here, the neuromuscular activity, which is seen by negative geotaxis assay, is much better when the flies are fed with CAPE, and similarly the stress response. All the other parameters are also better if they are fed with CAPE. Arpita has done these experiments on taking amnesia model mice, and she already explained to you, and the key results were that CAPE, and even better if the CAPE gamma CD complex is fed to those mice. So in amnesia or the memory challenged mice as well, CAPE behaved super here. So CAPE is good for memory. Now, in context of what we are all going through, COVID-19 pandemic, we know large number of infected numbers, and there is a lack of vaccine and drug. There's multi-targeted approach is required. Let me refresh you here about the virus infection cycle. They need attachment, penetration, biosynthesis, maturation, and lysis. And of course, we all know viruses do not have their own protein synthesis machinery. They use host cell machinery to replicate themselves. So here, Dr. Sundar in the earlier talk already explained to you this. On the cell surface, we have proteins called ACE2 and TMPRSS2, which act as host cell anchoring or the gate for the virus to enter. So we use these two, and we asked if the natural compounds we've been using in our lab for various purposes, for example, anti-cancer, neurodifferentiation, anti-stress, whether they would have any impact on these two proteins. And here again, the teamwork in the lab, Sukant, Jaspreet, Raj, and Priyanshu put their hands together, and they assayed about 60 compounds, which were actually identified as anti-stress compounds from various angles, and they are looking at expression level of TMPRSS2 in control and CAPE-treated cells. I didn't show you purposely the rest of the data because it's really in the process of making manuscripts, and some of it may be even patented. So what you can see here is that TMPRS2 protein expression is gone down to nearly 50% or 60% when the cells are treated with CAPE. So ACE didn't show. At the other end, again, the teamwork with the bioinformatics and computational analysis team in IIT Delhi, led by Dr. Sundar, and another PhD student, Vipul Kumar, has been very actively contributing these, actually initiating these projects. What he's showing here is that the CAPE has a capability to dock into the viral protein, which is essential for its replication. So it's not only the cell surface receptor, which is TMPRS2, but also the viral protein will be affected as good as its known inhibitor is. So here I'm showing you the docking for known inhibitor N3 and the CAPE, and you can see the same amino acids are involved in these interactions. So from my talk, the take-home message is CAPE has multiple activities. It's useful for managing stress at different phases of life, and age-related brain function is one of them, and the COVID-19 we all are going through. I guess I stop here, but one slide I want to show you here is honeybee propolis is nice supplement, but it is very sticky and it's got a pungent taste and it's difficult to take. So in collaboration with Cyclochem, we've been able to generate a powder of propolis, and we call it pleasant premium propolis, PP propolis. It also means P. PP is like in Hindi, you can drink. So that's PP propolis, which is not viscous, it's a powder, it is God stable, and it also protects CAPE from its degradation. I think this is my last slide, and I would like to thank you to all my students, postdocs, and the members of the group who have actually done the experiments, and special thanks to the team from Cyclochem, Dr. Terao and Ishida, who have been motivation for all honeybee projects in the lab. Thank you very much.
Thank you, Dr. Wadhwa, for very lucid and very promising presentation. So now this is open to questions from the audience. So if there are no further questions, sir, can we go on to the next presentation,
Sure.
please?
So Renu, can you log off from your presentation?
I will.
So our next speaker is Dr. Motomichi Doi. He is from National Institute of Advanced Industrial Science and Technology, AIST Japan, and he is a group leader. So he's going to tell us about simple experimental model system to explore the onset of neuronal diseases and their intervention by herbs. Dr. Doi, please.
Thank you, Dr. Kaur. So hello, everyone. Can you hear me?
Yes, we are. Yes.
Hello,
Yes.
everyone. I'm Motomichi Doi in AIST Japan. So thank you for giving me a chance to present our recent study in this symposium. So my talk is not so focusing on COVID-19 itself, but today I'd like to show our recent study to explore the potential of herbs for stress and the age-related neural diseases. So can we change my pointer? Okay. Okay, so as talked by former speakers, the recent pandemic of this COVID-19 is a big problem in our world. So in addition to direct danger of this virus, the COVID-19 is dramatically changing our lifestyle itself. So these social changes should generate a lot of stresses. I'm sorry, can I use this? Can I stop this pointer?
Sure.
So these social changes should generate a lot of stresses we have not had yet. So looking on the cellular level, several stresses related to COVID-19 can influence their cellular health. Of course, pathogens and virus infection can affect cellular health, but also the many other stress risk factors such as temperature and chemicals. So those stress risk factors can influence cellular health. In the cell, these stress induce misfolding of protein and changing the protein shapes. And the change structure probably induce the oligomerization of protein, and lastly, this oligomer can form large aggregate in the cell. So those are example proteins, they form large aggregate in and also out of the cell in our brain. And you may know that these aggregates are quite toxic to the cell and our brain, and so to be a cause of age-related neurodegenerative diseases. So we are looking for some effective natural components from herbs to protect from these diseases generated from the stress-related protein aggregations. So for this purpose, we are using a simple model organism C. elegans in addition to the culture cell. Those tools are quite useful to screen huge number of molecules quickly and easily. We have expressed these age-related stress-induced aggregated protein in C. elegans nervous system, and examined how these protein aggregate can affect the cellular mechanism. And also we can use these simple model organism to isolate the effective compound by checking the protein dynamics in living nervous system. So in the point of view, so we have found several novel cellular mechanisms against these protein aggregates. So in the case of alpha-synuclein, we found that the aggregated alpha-synuclein was transferred from postsynaptic neuron to presynaptic neuron through the certain type of synaptic connection. And also in the case of polyQ protein aggregated polyQ protein is exported from the nucleus by using the novel membrane envelope by nuclear membranes. So these exportation probably are quite helpful to save the nervous neuron health. And also in the case of amyloid beta, we have developed the novel fusion protein which can easily evaluate the aggregation state of A beta in living animals. Today, I will focus on this model amyloid beta system. So the keyword of our model system is visualizing aggregation state of causal protein. I think most of you are familiar with this famous Alzheimer's diseases. I will not have details of this concept mechanism. However, recent social change probably increase the risk of onset of this Alzheimer's diseases. So many mechanisms of pathogenesis have been suggested for these diseases, but the formation of senile plaques by aggregation of amyloid beta is one of the most causable, most cause of this disease onset. So you know that the amyloid beta is generated by the amyloid precursor protein APP by several enzymatic reactions, and the secreted from the cell to the out of the cell, and this small peptide can be easily aggregate out of the cell and form the large aggregate called senile plaque, and these senile plaques affect the cellular health and cause cell death in the brain. However, we have another argument against this classical amyloid hypothesis. So intracellular small amyloid beta oligomer is more toxic than the extracellular large amyloid beta aggregations and affect the several states of neurons. And those intracellular A beta oligomers probably are more toxic than extracellular large plaques. So to answer this argument, we need a simple and easy method to evaluate the A-beta aggregation in living animals. And this model system probably will be applied to isolate the candidate component or compound for Alzheimer's diseases. So, as you know, the fluorescence protein such as GFP is quite a useful molecule to visualize or to see the protein dynamics in living cell and animals. However, the simple fusion of A-beta and GFP does not express fluorescence signal because strong A-beta aggregation disrupts GFP conformation. So we have arranged the link sequence between A-beta and the GFP and found that the some link sequences can express GFP signal using the A-beta GFP fusion protein. So as you can see in this lower panel, if when we express the GFP only in culture cell, you can see bright signal in the cell as shown here. But when we use the short linker between A-beta and the GFP, you cannot see any signal in this panel. Also, we have enough number of cells in this disease. So I guess you are not so easily to see, but if we use a long linker between A-beta and GFP, the slight GFP signal can be seen in this disease, suggesting that this linker sequence can express fluorescence signal if the A-beta is aggregate in the cell. So when we introduce the mutation in A-beta, which inhibit A-beta aggregations, you can see bright signal in the cell, suggesting that the GFP signal is really corresponding to the aggregation state of A-beta. So we thought that this interesting phenomena can be used to screen the effective molecule or compounds suppressing A-beta aggregation easily. So, but you may think that the protein is really expressed in this kind of disease because we cannot see any signal in this disease. So we performed the native page to check the protein is really expressed in the cell and the fluorescence signal is corresponding to the protein dynamics in the cell. So you cannot see any signal in this disease, but we purify the protein from the cell and perform native page. So in this lane, you can see the radar-like signal as you can see in this lane too. So this means that the various sizes of oligomer exist in this cell. However, in the case of mutant form that suppress A-beta aggregation, you can see bright signal in this disease, but in native page, you just see only the single band, suggesting that the A-beta is expressed as a monomer in this bright disease. So we thought that our system is quite useful and quite easy to evaluate how the A-beta is in the cell. So we also applied our system to living animal in C. elegans. So we expressed our fusion protein in cholinergic neurons and examined how the GFP signal is. So as same with the culture cell system, when we express GFP only, you can see the bright GFP signal in both the cell body and nerve neuron axon. So as shown in this white circle and in white triangles. So those background signal is not the GFP signal itself, but the autofluorescence from the intestine. So when we use the short linker protein, so you can see any GFP signal in both the cell body and the axon. So suggesting that when the A-beta is aggregate, GFP signal is lost in the living neurons. So, but when we introduce the mutation, you can see the uniform GFP signal in the cell body and axon, suggesting that the A-beta is not aggregate in these cells. In the case of living neurons, when we use the long linker fusion protein, you can easily see where and when the A-beta aggregation occur in living animals. So you can see those bright dots in the cell body and also on the axon. So this long linker fusion protein can be used to examine when and where the A-beta aggregation occur in the living animals. So we have also applied our system to isolate effectively compound from using living animals. The transgenic C. elegans expressing our fusion protein are kept on the culture media, including the several chemicals or natural products. And we have examined the GAP signal expression in the living animals. So in the lower panel, so you can see one example using the curcumin, as you may know well. So when we use the short linker fusion protein in central nervous system, you can't see any GAP signal in the neural cell body and on axon. However, this one were treated by curcumin in 1 day. So you can see bright signal in cell body and on the axon, suggesting that this chemical can inhibit aggregation and the A-beta is monomerized by this curcumin. Okay, we will switch to our pilot screening using the phytochemical from the libraries. So we use HEC293 T cell as a model cell, and the A-beta GFP were transiently expressed in this cell, and directly evaluate GAP signal intensity from the cell. And the selected candidate were examined in a second and third screening. So here's an example of GFP expression pattern treated by several chemicals. So as I said before, the dark signal indicate A-beta is aggregate in the cell, and bright signal indicate that the A-beta is not aggregate in the cell. So in several chemical, you can see slight GAP signal in this panel and also bright GAP signal in this panel, suggesting that the A-beta is not aggregate, or A-beta is inhibited to aggregate by this chemical treatment. So we have performed several, we have used several concentration of chemicals and also several incubation time using the selected candidate. And we have found that these two chemicals have quite high potential to inhibit A-beta aggregations. So these two components show the dose-dependent manner to increase the GFP signal. So, but the signal is not enough as a mutant form, but we thought that these are a final candidate as a candidate chemical. So in magnification, magnified image, you can see bright signal by the treatment of this number 57 chemical. So this means that this chemical can inhibit A-beta aggregation. So this chemical is found in several seaweeds. So, and we are now looking for how this molecule can inhibit A-beta aggregation in the cell. And also we have applied our system to living animal, and we will skip this, I will skip this slide, but this number 57 chemical really has potential to inhibit A-beta aggregation in living animals. So as a conclusion, our screening system will be a quite simple and probably effective method to isolate candidate molecule from harm. And those molecule is probably candidate to affect intracellular A-beta aggregation in our humans. So those are members in our group, and I will thank these collaborators. Thank you for kind attention, and I'm happy to have a question.
Thank you, Dr. Doi. So we'll have a couple of questions if there are any, or comments. Well, if there are no questions, I on behalf of my co-chair, Dr. Simran, would like to thank all the speakers and the participants for attending this session. So with this, we come to the end of Symposium 13. Over to the organizer, please.
Yeah, very good evening again. And I'm really delighted and excited. First, to see you all and then to listen to you all. And all the lectures were very nice. Hi, Renu, and Simran, and Arpita, and Mitomichi. It was really a very exciting session. And you know how I like, especially the herbs. I really enjoyed personally. And I'm sure everybody else would have similarly really enjoyed, and I'm really feeling very, it's giving me a really very good feeling that across the globe, how we are connected, and so I'm really very happy. Once I had talked to Renu over a video call, I remember, but like this, I never thought we'll be meeting. Thank you so much.
Dr. Anita, I come in. I wanted to say something. Hi, Dr. Sunil and Dr. Renu. Very nice to see you. It was a wonderful presentation by your team. Just for the fun sake, how much ashwagandha we should take now to avoid COVID or to get rid of COVID? Yeah. Shall we start taking now?
Renu, your mic is unmute. You unmute your mic, Renu.
Yeah. Okay, so we are not the right people to really advise how much you should take, but I can tell you how much I take. I can tell you how much. I've got ashwagandha here on my desk. Yeah? I've got ashwagandha leaf capsules, powder, and I take 2 every day. And I know people who take even 10 every day. So it hasn't got any toxicity. So it's up to you to design your dose.
Oh, wonderful. That's wonderful.
Is it?
Yes, Dr. Prasad, good to see you.
Yeah, congratulations. A lot of things, conventional things. I mean, are you taking this ashwagandha in the form of capsule or in the form of powder?
It is actually the pure leaf powder. It is just, we've put it in capsule by ourselves because it's easy to just take it. So it is pure, organic, raised in Japan by, by very standard methods. And it is pure ashwagandha leaf, powder filled into capsule. This is how it looks like.
No,
This is how it looks like.
Yeah, okay, yeah, nice. Good. These type of products are available in the Indian drug market also.
Yes.
Do you know they are that good in quality also?
I have no idea.
I should not ask this, but just for my own confidence,
I have no idea what is sold in the market, and I wouldn't like to comment on that.
Yeah, certainly. I was not expecting to say any comment. Just for my confidence, I was trying to understand.
Professor Prasad, I know that even this organic India, they are also selling the ashwagandha capsules, and also organic tea. They are selling ashwagandha tea. So tea bags. And some people, I wanted to know a question from Dr. Renu, that some people, they say, why not simply pluck a leaf and eat in the morning? Some people prefer like that before eating anything. And they say that it's it's very beneficial. Can I have your comment on this?
Is there any experimental evidence on that? How
No,
much
no, I'm
dose? So we
asking
don't
you.
know. So
I'm
I would say, why not? If we are taking leaf powder, why not fresh leaf? But then fresh leaf is giving you much less amount. It's got about 80% water.
Yeah.
So if you want to have enough amount of bioactives, which are vidanolides, leaves are recommended as compared to the roots, which are sold in the markets. And we have shown that the roots have at least 20 to 30 fold less vidanolides. And if we are showing everything is driven by vidanolides, or at least all good things, so then we should consider that we are taking enough amount. Yeah.
Thank you.
Thank you.
Thank you.
Good to talk to you.
Thank you.
So
We can have a
congratulations.
distance. We can have a distance hug.
Yeah.
Yeah. Love you guys.
And to the Japanese friend, it is konnichiwa and konbanwa.
Yes.
Professor Kaul, I must convey my regards to you. I did not talk to Dr. Sunil Kaul. It won't be justified if I don't talk to both of you.
Thank you. Thank you so much.
I have been seeing both of you together, but this time, I mean, you are away in your country. I mean, in Japan, and we are in different places of India. So this is how we are meeting this time.
At least thanks to the technology, we can each see each other, we can hear each other, right? Still,
Yeah.
thankful.
And thanks to Anita also
Yes.
And
for bringing us together.
it is by virtue of his untiring effort. Dr. Anita
Thank
deserves
you.
for it.
Thank you so
For Anita, 10 tons and tons and tons of thanks.
हाँ
Yeah.
मैंने रात में भी आपने बहुत सारे भेजे हुए थे और मैंने जाकर सॉल्ट गॉगल्स किए थे
But आज आप टाइम से सोना.
In the chat
hi,
also I
ma'am.
wrote. Namaskar, madam. Namaskar.
Yeah, I would also like to thank Dr. Renita and the co-chair and the speakers. It was wonderful to be part of this symposium. I mean, learned a lot. So thank you, everyone. Thanks a lot.
Thank you,
Thank
Simran.
you, Dr. Kaur. Thank you.
Thank you.
Thanks, Dr. Kaur.
Thank you.
Thank you. Bye-bye.
नमस्कार.
Bye. क्या वो
आपका एम्स का IAN ऑर्गेनाइजेशन भी बहुत बढ़िया था. Congratulations.
Thank you.
Anji, thank you so much. Now, next time we look forward to Calcutta also physically meeting you all.
Let us hope that we meet there physically.
I have an announcement. The next
please.
session will also run on the same this thing because that's a plenary lecture and on the this login only. So those who will be attending plenary lecture, you can remain logged in here only.
Yeah, everybody would like to do. Yeah.
Yes. But that will start at 6. Slide out, yeah.
So this is a 15 minutes gap. Okay.
वो वापस में गप मारने
प्रसाद सर.
हाँ. Yes. How are you?
कल का सेशन हम बहुत लोगों से तारीफ सुने. आप अच्छा चेयर किए हमारे सेशन को.
Oh, this credit goes to you, and then it comes via you to me.
It comes to all the speakers. Actually, latest work was presented, so that was a beauty. That work was
For
published
me, it
just
was
now.
a new experience, and the organization, the organizers, I must thank them. They thought of me putting in the sleep session. So
And to keep you awake, and to keep you fully awake.
Yeah.
Okay,
So I like this topic. I had written to you that I love this topic. I teach this topic. I don't work, however, on this, but I learned a lot from the work presented, and your work was quite scintillating, and the others were too. So I mean, this is the kind of experience I had. This is unique.
Yeah.
Congratulations,
Thank you so much.
and thanks to you also.
Sure. Thank you very much, sir. Thank you.
Professor Patro. हाँ शुक्ला साहब।
नमस्कार प्रसाद साहब, कैसे हैं आप?
हाँ आप बताइए कैसे हैं?
सब बढ़िया।
चलिए लंबे समय के बाद हम लोग ऐसे मिल रहे हैं।
या
चलिए इसी को आमने सामने मीटिंग मान लेते हैं। It was very nicely done. In fact, I was not imagining it is. In fact, the credit goes to organizers, but also similarly it goes to the technology also.
Yeah.
तो और कहीं ना किसी को आना पड़ा न जाना पड़ा और सब लोग अपने अपने जगह पे.
And now we can meet at least 2 or 3 times in a year in this way.
Yeah. We have learned from this meeting. This is unique in its kind.
So whenever you want to just organize a symposium for 3-4 hours, 5-6 speakers, collect them and announce properly, and everyone will join. That's a good way.
Yeah, certainly. हाँ. चलिए आप कैसे हैं? सब बढ़िया है सब कुछ?
दुआएं हैं. Everything is.
हाँ चलिए.
Enjoying the
तो बहुत अच्छा यही वाला मीटिंग रहा है इसलिए कि मेरे लिए खासकर अच्छा रहा ये मीटिंग. यूनिक मीटिंग. मेरा एक्सीडेंट हो गया था तो मैं अगर फिजिकल मीटिंग होता तो नहीं जा पाता. और मैं घर पर हूँ और आराम कर रहा हूँ लेकिन मैं कॉन्फ्रेंस अटेंड कर पा रहा हूँ. ये अगर ई कॉन्फ्रेंस नहीं रहा होता तो शायद मैं इस बार एब्सेंट रहता. तो थैंक्स टू डॉक्टर अनिता दिस वे ऑल्सो. और आप लोगों से मुलाकात हो गई. नमस्कार. भाभी को भी नमस्कार बोलिएगा.
डॉक्टर पात्रो साहब कैसे हैं?
हाँ नमस्कार प्रोफेसर पात्रो. बधाई हो आपको.
धन्यवाद सर, धन्यवाद.
मैंने आपका मैसेज बैक हमको मिल गया. आई कंग्रेचुलेटेड यू.
जवाब दिया.
हाँ. तो बधाई हो. हम अगला मीटिंग में आपका इंस्पिरेशनल प्रेजेंस हम लोग देखेंगे. लेट अस मीट फिजिकली देयर.
Sure, sir. Certainly.
Let us wish and let us hope for that.
So there is a side by side. I have got a meeting going on with the Minister of Higher Education.
Okay.
I'll take 10 minutes.
Oh, please
continue.
Please.
Thank you.
Please continue. We are using our gap time to discuss and to know about each other.
Yeah, I'll come back. Just for.
Yeah. Okay. Fine. Okay. Yeah. Please go ahead. हेलो सर. नमस्कार.
why you are today saying all this?
Okay. Trying to call you. Is he there? Yeah.
Sir, aap le lo. Hello, sir. Namaste. Good evening.
Namaste.
Sir, you received, no?
Yeah, I received everything. Is Professor Gangadhar on now? Or is he?
Hello, sir. About to tell you.
Connected, Dr. Gangadhar. Okay, then you tell me when to start. Call karke and then you tell me when to start and then we start. Okay. Yeah, sure.
Namaste, sir. This is Shukla Prasad from BHU.
Aji, hello. How are you?
Namaskar, sir. Namaskar, sir. Well, sir. Well, by your blessings, best wishes. It's all okay here, sir.
Lovely. Is he connected? ठीक है, that's okay. Hello, we are just waiting for Dr. Gangadhar. I think he'll join very soon. We're just connecting to him. We'll get back to, we'll just continue here. I think I request everyone to continue, and we'll be here for a few minutes.
नहीं म्यूट हम
Hello, everybody. I think Dr. Gangadhar would be successful in logging in another few minutes. So please wait. We have contacted him and he's in the process of logging in. Actually, he has been trying. So,
हाँ 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 ठीक ठीक ओके फिर बाद में क्या कहेंगे ओ ओ ओके 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 ठीक हाँ 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 ठीक ठीक
Try to call BSS out to her.
वही वो कॉल कर रहे हैं. I have an announcement to make. Professor Shubhrat Sinha sir. Yeah, I wanted to make an announcement that due to some connectivity issue at their end, they're trying to log in. As soon as we get their connectivity, we will start the lecture. So another 5 minutes, sir. Sir, another 5 minutes.
Yeah, that is fine. We are waiting. That's
Yes.
Yeah, we'll all. Yeah. It'll possibly give other people in other symposia a chance to attend this talk, if the other symposia are still going on.
राकेश जी बहुत स्लिम लग रहे हैं.
मैंने कहा नमस्कार, आप कुछ व्यस्त हैं लिखने पढ़ने में.
Professor Gangadhar, good evening, sir. Namaskar, sir. How are you doing?
I'm fine. I sincere apologies. I just now came. I had gone out somewhere. I'm so sorry. I am ready with the talk, sir. Whenever you tell me, I am ready.
Yeah, he's come. Yes, sir. So can you please? So it is. Let me. Of course, Professor Gangadhar needs no introduction, but he is well known in the field of psychiatry and neuroscience for decades now. He is the director of the National Institute of Mental Health and Neurosciences. Also, its vice chancellor because it's a deemed university. Professor Gangadhar has been awarded the very prestigious Padma Shri by the Government of India and also the C.V. Raman Award of the Karnataka government. He is known for his, earlier on he was known very much for his work in biological psychiatry, and today he is in a way talking about, I suppose, the merger of biology with yoga. His topic is neurobiological markers of yoga used as therapy in mental illness. So Professor Gangadhar, over to you, sir. Please start your talk.
Thank you, sir. Thank you very much. At the outset, I sincerely apologize for having been very late. I'd gone out somewhere and there was no connectivity and so on and so forth. And I was just able to catch up with all of you. Once again, I profusely apologize. Today's talk is going to be on neurobiological markers of yoga therapy. Now that I have gotten delayed, should I bring down my talk to about 30 minutes, sir? I want the chair's permission, whether I have that earlier 45 minutes or probably not, because I have been late. I have to pay back that time back to you. So I'll try to wind it up as early as possible so that the entire session won't get changed. Ever since the declaration of the International Day of Yoga by UN, yoga as a formal event, it has become terribly popular, and all over the world people are practicing yoga, and no wonder the research contributions in yoga have also gone up steadily. And today I'll be speaking to you about the biomarkers of yoga therapy. The topics I'll cover include depression and schizophrenia. These are the two areas where biomarkers in yoga therapy have been fairly well studied, and most of it has come from our laboratories. I'm going to share that data with also some data from other laboratories as well. The question that we have posed in all our research is, would yoga work on just symptoms of the disease, or also on the pathobiological processes, and thus being a cure? Although a very sensational question, I want to demonstrate what are the biological markers of therapy in these two disorders. Let's start with depression. I used depression as an example because it is one of the most common psychiatric disorder, ubiquitously affecting all ages and either sexes. Although more females at any point of time are found to be depressed than males, but the point remains that both sexes are affected at all ages. We began with an initial randomized trial of yoga in depression, and we were able to demonstrate that yoga works, Sudarshana Kriya works as well as antidepressant drugs in the treatment of this disorder. It was a randomized study. We also compared with ECT. ECT was better than either of these two interventions. And there, of course, more recently, people have published on yoga as a monotherapy in depression, also finding yoga is effective. Most recently, we did another study where it is not a comparative study, but suffice to demonstrate that yoga brings down the depression scores in people with outpatient treatment for depression. Even when yoga alone was given, the reduction in depression scores was substantial. I don't want the listeners to see this slide as a comparison between yoga and antidepressant drugs, because this was not meant to be a comparative trial. These are not randomized. These are all patient-selected samples. A meta-analysis also indicates that yoga is better than usual therapy. Yoga is better than relaxation. Yoga is better than aerobic exercise in the treatment of depression. This meta-analysis is a strong proof that yoga works. Now, let me illustrate what are all the biological processes which are detected in individuals with depression and what happens with the yoga treatment. Take, for example, the brain physiology. Depressives are known to have lowered frontal cerebral gray matter activity and a corresponding increased deeper structure activity that includes amygdala, hippocampus, anterior cingulum, and so on. So these limbic cortices, limbic gray matter is hyperactive in depression. And depression is also associated with reduced levels of GABA activity, whether in men or women. GABAergic transmission in depression is lower. Depression is also associated with elevated levels of cortisol, well demonstrated. Although we know that the cortisol levels in the individual vary, if you do a multi-sampling method, levels of cortisol is higher in depression demonstrating that there is a stress effect of depression in these individuals. Depressives also have lowered levels of brain-derived neurotropic factor. Lowered brain-derived neurotropic factor is there in patients compared to healthy subject. What you see on the y-axis is the effect size of the difference. It becomes less when you compare after treatment and the healthy subjects. After treatment, depressives would reach values closer to that of healthy subjects. And after treatment, the depressed individuals also have elevated levels or higher levels of BDNF than what they had in treatment before. Chronically depressed individuals also have lost gray matter in cerebral cortex in different areas as illustrated in this slide. As we say, Chitta Dahati Nirjeevam, Chinta Dahati Jeevanam. Chronically being in Chinta makes them lose their gray matter in the brain. This is a culmination of effect of increased cortisol, lowered levels of BDNF, and then leading to lowered levels of gray matter in the brain. And what does, this is the cycle of events that happens in depressed individual, increased activities of the amygdala, which comes from the decreased frontal activity, decreased GABA activity because of the increased activity of the amygdala, increased cortisol levels, decreased levels of BDNF, and cortical thinning. This is a vicious cycle that happens in an individual who is depressed. And what does yoga do? Yoga actually affects the brain physiology at all these levels. Let me demonstrate this in this study that we have done at NIMHANS. For example, what happens to the physiology of the deeper structures, which are actually hyperactivity in depression. This is in healthy subject. We did not do it in depressed individuals, but in healthy subjects, when they are practicing Om chanting. Om chanting is a part of most yoga practices and yoga modules. Even in depression module, in NIMHANS Om practice, Om chanting is a part of the depression module for yoga. Deeper structures become deactivated. Anterior cingulum, another limbic structures are deactivated following while they were chanting Om, as compared to when they were chanting a non-Om his sound. So one of the practices actually reverses the or produces a physiological condition which is conducive to reversing what would have otherwise happened in depressed individuals. We looked at what we call as the cortical silent period in transcranial magnetic stimulation. You give a transcranial magnetic stimulation single pulse to stimulate the thumb muscle, which is actually in contraction with high EMG activity. Zoom, the EMG activity goes up in the same area. Following which there is a short period of silence in the EMG, which is produced by the cortical silent period mediated by the GABA activity. And then the EMG resumes. So this length of the length of the silent period in the cortex is the cortical silent period, which is related to the GABAergic tone of this individual. What happens in depression? Look at the pink one on the left side and the yellow one on the right side. Yellow one is a healthy subject. The pink one is on depressed individuals. Outpatient depressives have shorter length of the cortical silent period, indicating they had lower levels. And you allocate them into either walking or yoga practicing as a form of intervention in their depression. Walking also produces reduction in the depression scores, but the GABA activity as measured by the cortical silent period using TMS does not change. Whereas that of depression, the light blue and the dark blue graphs indicate that in depression individuals, the GABA activity went on to get longer. The cortical silent period went on longer, inching towards that of the healthy subjects. Elsewhere, people also have examined the direct measurement of the GABA the GABA signal in the brain measured by the magnetic resonance spectroscopy went on to get taller in individuals when they practiced yoga. A yoga session acutely elevated the GABAergic levels in the brain, suggesting that the yoga does reverse the GABAergic pathology in depressed individuals. Next, the cortisol levels are higher in depressed. We have been able to demonstrate in depressed individuals who are attending the outpatient clinics. You give them medications, yoga with medications, and yoga alone. Mind you that this is not a comparative study. Only those available for follow-up, we were able to examine pre-yoga and the post-yoga cortisol levels. Cortisol levels got lower in higher number of subjects when they practiced yoga, either with medicines or when they practiced it alone. Their depression scores also got better. We also found that the reduction in the depression scores correlated with the elevations in the with the reductions in the cortisol levels as well. So lower the cortisol, the better they were following yoga. So in other words, yoga reversed the hypercortisol levels of depression, which was observed in depression individuals. Coming to BDNF, you see the two blue bars on the left side. One of them is that of the patients of depression, outpatients attending the treatment at NIMHANS before they started any treatment, and the healthy subjects, the taller blue bar. They were different, and this was significantly lower in depression. You treat them at the end of 3 months of treatment, the saffron bar, which is a slightly taller one, the depressed individuals went on to have increased levels of BDNF. This group of depression was that of those who received depression, those who receive antidepressant drugs, or with yoga or yoga alone. Even when you examine the groups separately for yoga alone or yoga with medicines, significant elevations in the BDNF level were demonstrable with practice of yoga, either alone or with antidepressant drugs, suggesting that yoga increased the levels of brain-derived neurotropic factor. Very interestingly, we found another association in people who lost the levels of cortisol, those who went on to get decrease in the cortisol levels following the therapy with yoga. In that group, the BDNF level went on to rise, but not so in the group where the levels of cortisol continued to remain high. And in this smaller sample, 11, the BDNF level actually did not go up. In fact, it even went down. In another group of healthy elderly subjects with mild cognitive impairment, when yoga was practiced for a period of 6 months, this is a small sample of 7 subjects where we were able to measure the volume of gray matter using the statistical parametric mapping, where we, in the hippocampus, that is the only area which we measured, before and after, there was a small but significant increase in the cortical gray matter in hippocampal area, suggesting that yoga in the elderly, this was the average age of this group was 70 years, and then they practiced yoga for a period of 6 months under supervision. And before and after, when we examined the MRI as a comparison using statistical parametric mapping, significant increases in the cortical gray matter in the area of the hippocampus was demonstrable. Limitation is that we don't have a control group which did not practice yoga, but in the elderly individual, we thought there would probably no reduction in the cortical this gray matter of the hippocampus, but what we found was the gray matter in the hippocampus actually went on to increase. This could well be an effect through other mechanisms which we described earlier, but given that this group was elderly, they did not have depression, limitations and extrapolation to depression can be a matter of question. However, there are other people who have demonstrated that the total gray matter volume reduces with in healthy subjects with age. This age-dependent reduction of gray matter was not seen in individuals who had been practicing yoga for several years. Again, corroborating, although in a cross-sectional data, that yoga produces protection against neurosenescence. We also have physiological markers of increasing in the event-related cognitive potential, P300. When yoga was practiced in depression, depressed subject had lower amplitudes of the ERP, and the ERP amplitude event-related potential P300 went on to increase following yoga as the intervention, sole intervention. So I'm recalling the cycle of event that will happen in depression, and I'm demonstrating to all of you here that yoga intervention increased the frontal activity, although we don't have the data from our study. Elsewhere, it has been demonstrated. And the hypoactivity of the limbic cortices have been demonstrated, at least with one yoga intervention, leading to lowered or increased levels of the GABAergic activity, reduced cortisol levels, increased the brain-derived neurotrophic factor level. Both of them have an association though. And then prevention of the gray matter loss, at least in a group of healthy elderly subjects. So at all levels, the yoga effects are demonstrated, again suggesting the fact that Chitta Dahati Nirjeevam, Chitta Dahati Jeevanam is reversed by the intervention of yoga in depressed individuals. I'll quickly allude to similar findings of neuromarkers, neurobiological markers in schizophrenia. In schizophrenia, yoga has been effective. We used schizophrenia as another model because schizo means splitting of the psyche, thought and emotions are disconnected, emotions and action and so on and so forth. And schizophrenia individuals have a host of psychological symptoms which have posed challenges in treatment. And it is one of the condition which is also manifested by social cognition deficits, which are a bugbear and they don't respond to treatments as well. Schizophrenia studies, the neuromorphological studies also indicate a biological schism. The corpus callosum is pathological in schizophrenia individuals. So together, the disconnection in schizophrenia ought to become connected with yoga, because yoga means to connect. Samyoga yoga ityukto jivatma paramatma, merging one's personal consciousness with the universal consciousness or the cosmic consciousness. So this spiritual journey to the ultimate destination of connecting to that universal consciousness should also have a forerunner of connecting to other consciousness and connecting within oneself. This ought to happen, and so the individual ought to get benefits from yoga in schizophrenia as well. We have done a couple of studies. One of the recent ones which we have published is a randomized study where we examined yoga, exercise, and a waitlist group in chronically ill schizophrenia individuals who have been stabilized with antipsychotic drugs and coming for follow-up in the outpatient, yet continue to have significant morbidity in terms of negative syndromes being quite high. In these individuals, when yoga was given, and when you compare yoga versus exercise versus waitlist, yoga produced significant reductions in the negative syndrome. So more people with who got yoga lost or had a reduction in the negative syndrome than those who received exercise or just were waitlisted. And in fact, we also were able to demonstrate that the social cognition tests went on to get better following yoga intervention as compared to a waitlist. In waitlist situation, the social cognition scores did not get better after the period of about 3 months of study. And whereas in 3 months of study, the social cognition measurements had gotten better in these individuals. We recently published these data, although at the time of making the slide had used only the data from the ongoing study. There are examples to illustrate with case history that yoga makes this connectivity or connectedness much better following in schizophrenia individuals. One of the patient's mother told me that, doctor, you gave back my son to me. I'm not going into the details of this case history, but the fact remains she noticed that this individual, after about 1 year of yoga therapy, although he was been getting antipsychotic treatment for almost 7 years, this last 8 months of introduction of yoga, he had several changes which made this mother feel that he was more connected to this individual. And this connectedness as an experience has been reported by many other studies in depression as well. And even in healthy subjects, connectedness is a common experience following yoga. And if you see one of the third commonest experience which is reported by individuals who are practicing yoga is that they feel connected. So I believe before connecting oneself to that universal consciousness, one starts getting connected to the neighboring consciousness and gets this experience of feeling connected. These studies from NIMHANS on yoga in schizophrenia influenced the NICE guidelines to include yoga as a part of urgent therapy in chronically schizophrenic individuals in outpatient management. So yoga effects are mediated through the oxytocin and the mirror neurons. We have data that mirror neuron activity is lower in individuals of schizophrenia and tends to get better following yoga therapy, following antipsychotic therapy. And we also found that the mirror neuron activity have a relationship with negative syndromes of schizophrenia. And there is also this is demonstrated very well by functional neuroimaging studies. And we also had some initial data which we couldn't confirm in subsequent studies, was that the mirror neuron activity went on to increase following yoga. In a healthy subjects, the same we were able to demonstrate as a replication in schizophrenia individuals as well. The mirror neuron activity could be measured by typical tests, and we used functional NIRS, near infrared spectroscopy, to illustrate the phenomenon in individuals, healthy individuals, and also in schizophrenia individuals, where the activation of the proposed or putative mirror neuron center got brighter after the yoga practice. This is something that we were not able to confirm in repetitive studies. Oxytocin levels have been found to be lower in schizophrenia in other studies, and we also were able to demonstrate, or we found other studies that lower levels of oxytocin were associated with lower social cognition. We gave schizophrenia individuals yoga and compared them with the wait list. Schizophrenia individuals went on to have increases in the levels of oxytocin, but not so in the waitlisted group. And the group which went on to get yoga and had increases in the levels of oxytocin also obtained improvements in social cognition. These observations also are supported by yoga studies in autism, where yoga applications went on to increase imitative responses in these kids who were given. The same thing, where people are now using nasal sprays of oxytocin to get the social cognition better. And we here have oxytocin autogenously elevated following yoga practice, leading also to the same effect. Lastly, regarding connectedness, in the brain, connectedness is lower in schizophrenia individuals, and we have seen the default mode networks coherence is lower in schizophrenia subjects. And following yoga practice, the individuals went on to get better with respect to their negative symptoms, also the social cognition effects. And the posterior and the anterior default mode networks were less coherent before treatment and went on to become more coherent, as you can see the brighter images using the functional magnetic resonance imaging. So yoga, as we say, connects the individual, gets better from schizophrenia, and these effects could be because he is mimicking the other therapist, performing them with mindfulness, paying attention, synchronizing one's breathing to the movement. And the individual also gets his mirror neurons better, needs to be confirmed in subsequent studies, and increases in the oxytocin levels and also the GABA mechanisms as seen in depression studies. Together, social cognition and connectedness get better in schizophrenia individuals, and they get better. So together, we have biological markers of yoga therapy in depression, increases in the levels of the BDN, reductions in the levels of cortisol, increases in the levels of functioning of the GABAergic tone, and the lowered functioning of the limbic structure, depression, and then, of course, prevention of gray matter loss. Together, this cycle of events reverse what would otherwise happen in the pathobiology of depression. In schizophrenia individuals, increasing the levels of oxytocin and increases in the DMN connectivity to make the schizophrenia subjects better. Stop here, and I thank all my colleagues who work with me. I also thank the IAN for having invited me for this meeting, and this is the email address to which you can send any questions if you have. Once again, let me thank Dr. Shashi Bala Singh, who sent the invitation to me. And
हमारी शक्लें दिखा दे निशु. नहीं वो हटाएगा तो हम.
I think Dr. Anita, mute yourself, please. Sorry. Yeah. Sir.
Thank you, sir. I'll close my slide sharing feature so that I can come back on the screen.
Sir, thank you very much for this very enlightening talk. I think you managed to compress so many years of experience and decades of experience in such a short time.
Thank you, sir. Thank you very much.
So, what is interesting is that many of us still do not associate the biological with yoga. And of course, there is also a community or, in the general society, biology with psychiatry, again, many people don't make that link. And many people don't make the link between biology and yoga. What you have done is actually shown the biology as reflected by, functions of the brain being connected to both psychiatry, mental illness, and yoga. And for this wonderful synthesis, IAN is very grateful to you that you could present it to us. Now, sir, I think a few questions, if from the audience, would be welcome.
I'll be happy to answer,
some 5-7 minutes more if somebody would like to ask or, make a comment or discuss briefly.
Yes.
The questions are in chat also, so you may.
Yeah, I have seen the questions.
Okay.
Can I ask a question?
Okay, Anita, please.
Okay. Sir, very good evening again. And sir, this is Anita. I was ringing you all the time. And I really enjoyed your talk. And I was really very happy to see how you have correlated the diurnal rhythms with the yoga and medication. So I wanted to know, sir, like how these biological clocks, especially the sleep-wake cycles, are being regulated with this meditation practices.
Madam, I have to admit that I'm not a physiologist, not a yogi. I have had a very focused application of some physiology and some yoga in mental illnesses. Biological clocks is a vast subject, as you know, it even got the Nobel Prize. I must admit.
I work on biological clocks, that's why.
Madam, you ought to know. But I believe I can only surmise. I won't be able to give you the answer if there is an answer. Anybody has done that. Yoga harmonizes. What is increased is brought down. Samatvam yoga uchyate. That's what is said. What is increased gets lower. What is reduced are brought up. So this equanimity, this caused by yoga, is probably also linked to its harmonizing its effect on the biological clock. The clock, if it is running fast, it will run slower. If it is running slow, it gets faster. This is a very, very simplistic answer I can give. I have myself not done some work on this. The closest work I have done is the experiences of sleep in elderly individuals who have been practicing yoga. We have published this article though. Sleep experiences are better in individuals who have been practicing yoga. Beyond that, I may have very limited knowledge on this subject.
Sure.
There is, I'm just reading out one of the questions which has been there. Well, I think I'm synthesizing a few of the questions which have come in the chat box. One is the differences of biological of response to yoga amongst age groups, genders, and types of yoga. So this is a very vast question, but brief words, if you could briefly tell us.
Is it right that whether yoga works in different groups of individuals?
Is there difference
I have to say
in
that
the response to age groups? Are there gender differences in response? And are differences related to the type of yoga practiced?
Again, sir, the research work on yoga in using biological markers has had a very limited approach. Not many centers and not many universities have been doing it. But from whatever I know of yoga effects in general, have been profound in both sexes. There are no difference. Would men have better response than women? There is no data to that effect, best of my knowledge. And yoga effects have largely been studied in adult population, although only a small proportion of studies have focused on children. So most studies on yoga having been done on healthy subjects who are adults, we can only extrapolate it to adults. The extremes of age, both children and elderly, the studies are not really many. But in all the groups, biological changes of different nature have been demonstrated, depending upon the nature of experiment that they were doing. There have been some questions on telomere length.
Okay.
Madam,
good evening, everyone. And I must congratulate Dr. Gangadhar for an excellent talk. I am Rakesh Shukla from Lucknow. I have a very quick question. The physical exercises of a yoga and the respiratory pranayam kind of exercises of yoga, which are more beneficial for the mental disease like a depression or schizophrenia or this kind of thing? That's my question.
Yes, sir. We examined.
So can I extrapolate this a little bit more related to some of the questions? Are there different yoga modules prescribed for depression or schizophrenia? And what is the basis for that if we were to extrapolate this whole issue?
I wish I had an opportunity to show you another slide. We have, in all yoga researches that we have done, we developed what is called as a yoga module. Yoga module development has some steps. Number one, we look at the traditional textbooks of most commonly practiced yoga and most easily doable and the yoga practices that are simple to teach. A list of yoga practices and their effects as has been described in traditional texts. We make a big list of them. Please remember that yoga books are not pharmacopoeias. They are meant to describe the way in which we do yoga and what are the benefits that each yoga practice gives. And then we have a given syndrome, let's say depression or schizophrenia or somatoform disorder or ADHD and so on. For each condition, we have a list of most commonly presenting features, clinical features. And then we match the descriptions of the yoga benefits and the clinical features, which are the ones which the yoga practices actually counter. Based on that, we have a decoction of yoga practices, maybe 6 or 7 simple yoga practices, which are put together in an order which the yoga therapist finds it most acceptable. And once this module is drafted, we send it to yoga therapists who have been practicing yoga for several years for validation. they give some suggestion that the order in which the yoga practices should be different, or you add this one yoga practice, maybe you have missed out in your literature research. And this yoga practice is generally useless and causes more harm. So together we generate a yoga module, which is called as a generic yoga module, and we run a pilot study of using this module, feasibility and what we may find as beneficial effects and some safety effects in a small sample of pilot data. And once we are sure that this is safe and it is feasible to teach them and they report some beneficial effects, then we go on to put this yoga in a clinical trial, either just comparing this with and then look at the biomarkers in this study. This is the cycle in which we go up and develop, whether in depression or in schizophrenia. This is the way in which we went on to develop. In depression, yoga is practices of yoga are better than merely walking. Walking has been recommended as an intervention to get mood relief. So we have used walking. In schizophrenia, we have found a group of exercises which are taught in high school students. Exercises have been recommended in schizophrenia individuals. So there are, of course, a lot of studies, and we have also reported an exhaustive review comparing yoga and physical exercises and their clinical effects and the biological effects in healthy population and also in different clinical subjects as well. This review is available on the net, comparison of yoga and physical exercises by one of my colleague, Dr. Govindaraj, which is well cited, and you can always access it on the Google itself. The journal allows a free download for the article. So yoga effects have been compared. Yoga effects, the way in which developed yoga modules are different. Each disease has a yoga module that we have developed. Although the difference between a yoga module of schizophrenia versus depression is definitely there, but it is not very wide. Three fundamental principles operate in all our yoga practices. One is the physical posture, Sthiram, Sukham, Asanam. And then we always synchronize all yoga practices with the corresponding breathing. So a person becomes mindful. And we encourage the individual to be mindful about all yoga postures and the yoga movements dynamically he makes to reach the posture. So continuously from the time he starts the yoga practice towards the end, expected mindfulness is also added into the whole process. So movements, breathing pattern, and mindfulness, they determine the effects of yoga in these subjects.
Sir, thank you very much for this very wonderful tour de force. Now, with this, I am sorry to have to conclude the session because our next plenary starts at 7 o'clock and it is 6:59. And much as I would have loved to let you go on and on, I think and wonderfully, concisely wrapped up the essence of yoga. Professor Gangadhar, thank you very much.
Thank you, sir.
I have a question.
There's a question.
But just one question.
Okay.
Only one. Sir,
Last
very
question.
nice talk. Myself, Dr. Prachi, sir. It is very informative. But I have one question that most of the young generation, they wake up late in the morning, including my children too. So does yoga affects with the time also that in morning yoga is much beneficial rather than in the evening time yoga?
Please tell that to your children, madam, to ask them to do yoga in the morning. They will get up early in the morning.
That's I'm telling sir. That's I'm telling sir. But it is a very general problem of young generations. That I'm telling.
Yes,
you. So morning yoga is more beneficial. Because
no data.
No data. Okay.
Okay,
Thank
sir.
Thank you so much.
Thank you so much.
I'm again sorry, sir. I joined late because of my predicament. I apologize to the entire audience and the IAN team for making all of you wait.
Thank you. You more than made up for it by your wonderful talk.
Thank
and we are unwilling to let you go, but we are forced to let you go now that our next session will start. Thank you, sir.
Thank you, sir.
I think.
Thank you, sir. Thank you, Sinha, sir.
Thank you.
Thank you, Professor Sinha, sir.
Thank you very much.
And I have some announcements, sir. Hello.
Hello, yes, please go ahead.
So the next session on technical lecture by Hiroke R. Oeda will continue on this link, and the panel discussion will be on the second link.
Okay, so all of us who are going on to the panel will move off and others will move on. Thank you. Yeah.
Thank you. So I now invite. Time है ना? हाँ. निशु मेरे को बड़ा कर दो. So I now invite chairpersons Durai Sundar and Dharmendra Khatri to chair the session. Can I see Hiroke? No, I want to say hello to him.
Yes, can you?
I can't
Hi.
see. Hi, Hiroke. How
Yeah.
are you?
Great.
Great, great. Good to see you.
Good to see you again. Yeah.
Yeah, very nice. And you always adjust time. so nice. I was worried. So I take pleasure in introducing the chairperson, Dr. Khatri, Dharmendra Khatri is here, and
Yes,
and Dr. Durai Sundar may be in the process of joining. So I can introduce first Dr. Dharmendra Khatri. He is currently working as faculty in Department of Pharmacology and Toxicology at NIPER, Hyderabad. And his specific interests are multidisciplinary approach to investigate the cellular and molecular mechanisms underlying pathogenic degeneration of specific neuronal population in the PD brain. He has many recognitions and awards and fundings, and he is also Indian ambassador on behalf of International Society of Neurochemistry and is next generation young neuroscientist for Basic Science Summer School, which was held recently in Osaka University, Nakanoshima Center in Osaka in 2018. And I will briefly introduce Professor Hiroki and leave the platform for the chairperson. I personally would love to introduce. That's why, Dr. Dharmendra, I'm taking over.
yes, ma'am, please.
Because I know Hiroki, as all of us call him Hiro. So that's a short name. So Hiro, I am meeting him in most of the clock meetings, especially, and in the neuroscience meeting at international platform. And I always enjoy his lectures. So I thought everybody in our conference also should listen to him and get that feast of his lecture. And he is professor of biology at University of Tokyo and the Riken Quantitative Biology Center. His specific interests are system biology and focuses on chronobiology by investigating mammalian circadian clocks and sleep-wake cycles. He has a number of honors and awards and publications in high-impact journals and well-established, outstanding neuroscientist, but I will just name few. He's a Tokyo Techno Forum 21 Gold Medal awardee, and Young Investigator Award and IBM Science Award in his kitty, Innovator of the Year 2017. With these few words, I do not want to encroach onto his time. I over to Dr. Khatri to invite the speaker.
Thank you very much, Dr. Anita ma'am. And now we have a a very interesting lecture with the Professor Hiroki, and all the audience are so excited to listen your very interesting talk. Sir, please welcome and start your talk.
Can you see my slide?
Yes, sir, absolutely.
Yes, okay. And thank you for inviting me. Especially, I'd like to thank Anita and also Dr. Khatri and other colleagues in this symposium. So today I'm going to talk about the whole body and the whole brain imaging with single cell resolution by using a new tissue clearing technique called QBEC. And then, as Anita nicely introduced me, please call me Hiro. Okay. But I just before I introduce our technology, I like to introduce my motivations. So, technology is a technology to understand something, right? So we develop this kind of a technology to understand the molecular mechanism or cellular mechanism underlying mammalian sleep-wake cycle. And then I briefly introduce our recent work on non-REM sleep and also REM sleep. So in REM sleep, we recently identified the first essential genes of REM sleep. So if you abolish just two genes like M1 and M3 acetylcholine muscarinic receptor, REM sleep in the mammalian sleep-wake cycle completely gone. So this is a kind of a first report on non-REM sleep essential genes. And also regarding on the non-REM sleep, we found first sleep promoting kinases, which turn out to be a CAM kinase to alpha and beta. And then if you, knocked out these genes, you observe the sleepless phenotype in mammalian sleep. And then when we overexpress these kinases in dominant active form, we can increase the non-REM sleep, meaning these genes are really involved in the non-REM sleep regulations. And then if you're interested in these, work and also background, we recently published two, review paper. First on the REM sleep, which is published in 2019, and also in 2020, couple of days ago, October 2nd, we also published a paper on the hypothesis in which phosphorylation cycle might be underlying the non-REM sleep regulation. So if you're interested in the sleep or wake, please check it out. Okay. So to understand sleep-wake cycle, we like to look at the brain activity. To that purpose, we recently developed a tissue clearing technique, which allow us to look at every single cell existing in our brain. And here is one of the state of the art. So in 2018, we succeeded to clear the adult mice brain. And then we try to look at every single cell existing in the adult mice brain, which turned out to be 120,098 cells in that particular brain. Okay, and also we recently succeeded to look at every single cell existing in the body of the mice infant, and then which turned out to be 428 million cells in the infant mice bodies, which is not published yet, but that is kind of a state of the art in this field. Okay. So for the beginners, if you don't know that tissue clearing or whole organ imaging, please look at my recent review, which is both published in 2020, I mean this year's, from Nature Review Neuroscience and also Neurons, okay? And then, so how, why, your organ or mice organ become transparent? Actually, tissue clearing field is initiated almost 100 years ago, okay? And then German anatomist, Professor Spartehote, proposed two basic physics principles. So one is to reduce light scattering here. Okay, so you can imagine there are many substances in the cells. For example, proteins or lipid or waters. Okay, and then each substances has a different property for the light. Okay, so for example, in the water, light travel faster than other substances. Okay, and then so therefore, if the light goes through the many different substances, it cannot go straight. It scatters, okay? So to avoid such scattering, we try to remove the lipid, and then we try to adjust the refractive, property of the light in the certain substances. For example, we try to manipulate such property called refractive indices, and then match them with the proteins. Okay, so and then we can allow the light go straight. Okay, so and then another issue is absorption. Okay, so in our bodies, so there are many pigments, like a heme in the red blood cells, okay? So to remove the heme, we usually, perform the perfusion to get rid of the red blood cells. But it's usually difficult. So because, in the very tiny blood vessel, red blood cells still remain, okay? So therefore, we sometimes use a very strong chemical. But that chemical, of course, destroy the pigment, but also destroy the protein. That's the problem. So therefore, we have to deal with these issues. Okay. So far, there is no good technique for to reduce the light absorption. Okay. Okay. So and then, as I said, this field is initiated by Professor Spartehote almost 100 years ago. And then he used hydrophobic reagent, meaning water insoluble reagent. Okay. And then many years ago, almost 80 years ago, 80 years later, Professor Tudin in Russia tried to introduce the hydrophilic reagent into this field. Okay. And then so far there are three categories of this. Three categories of method in this field. So one is hydrophobic, the other one is hydrophilic, and the third one is hydrogel-based method, which is recently developed by US researcher Casey and also Carl Daiselos. Okay. So there are pros and cons among these three categories. And we are focusing on the second category because hydrophilic reagent is safe, and also it can preserve the fluorescence proteins, and also it's easy to use. And then only problem was a little bit low performance. Okay, so therefore we decided to improve this performance by introducing the chemical screening strategy into this field. Because we have already 100 years, okay, in the research history, and then we wonder, how can we contribute to this field? And then we try to bring some kind of randomness by using chemical library, and hopefully to introduce the serendipities. Okay, But it's very difficult because if you want to use one organ for one compound, you have to sacrifice many animals, which is not good in terms of the animal ethics, and also they very labor intensive. So therefore, one of the undergraduate student come up with a new idea, and then he smashed the brain and then fixed the brain to make a brain paste and to use for the chemical screening, and it works pretty well. So we tried to measure the transparency by using organ itself, and also by using brain paste, and then we found a very good correlation between these two, which mean that you can use brain paste for chemical screening. And then we tested bunch of different compounds, and then we found couple of chemical called amino alcohol is quite key in tissue clearing, especially for the removing the lipid. Okay, and then we combine these chemical with other detergent as well as urea to come up with tissue clearing cocktails, and then apply them to the adult mice brain. And then, as you can see here, mice brain become very transparent. Okay, but interesting things happens because when we apply this technique not only to mice brain but also to the mice body, it can also work pretty well, which is accidentally identified. Okay, so one of the undergraduate, I think graduate student in neighboring lab using our reagent, okay? And then they tried to put the blood-rich liver into our reagent, and then they said it works pretty well. And then I was so surprised because we already know red blood cell is not good for the tissue clearing, okay? But they said it works pretty well. And then that was at night, and then I went to their lab and then tried to look at their sample, and then it looks very transparent. And then, but we noticed colorless tissue clearing reagent become yellowish colors, and then yellowish color turn out to be a heme, which is eluted from red blood cell in the later stage of the research. Okay, so, and then we tried to investigate these things, and then we found amino alcohol, which was screened by chemical screening, was usually turned out to be a potent chemical to elute the heme. Okay, and then the underlying mechanism is something like this. So heme and the hemoglobin are interacting through the nitrogen bond, and then amino alcohol nitrogen may be able to compete with the interaction between heme and the hemoglobin. Therefore, they can elute the heme in very mild conditions. Okay, which I like to touch upon the later stage of my talk. Okay, but by the way, we can apply this technique to the entire body. Okay, so after you clear the organ, you can use a microscopy, kind of a specialized microscopy. So usual microscopy is something like this. So illumination axis and also observation axis is usually horizontal. I mean, it's the same axis. Okay, but in this specialized microscopy called light sheet microscopy, illumination axis and then illumination axis and observation axis is a vertical. Okay, so you can illuminate from the side and then observe from the top. Then if you can change the height of the illumination, you can change the image, okay, and then you can stack the image to get the three-dimensional information of the organs. Okay? So by using this technique, you can get the whole organ image of the heart and lung, and then liver or kidney, okay, something like this. And then this is a kind of a three-dimensional anatomies. And also you can apply this technique, not the anatomy, not the anatomy itself, but also to the pharmacologies. So here is an example of the schizophrenic mice. Okay, you can apply. So in this case, we administrated the NMDA receptor inhibitor into the mice. Okay. As you may know, NMDA receptor inhibitor are known to induce a schizophrenic symptom in the humans. Okay, and then here is a mice, and then because that the mice, so therefore you cannot ask them what can you see or what can you hear to check the positive symptom of the schizophrenia. But as you can see the mice brain, you can see the entire cortex is highly overactivated in the administrated mice, as shown in here by red green fluorescence. Okay. So auditory cortex and then visual cortex is highly activated, meaning mice may hear or mice may see something. Okay. All right. So that was a 2014 situations. Okay. So we found very potent chemical, but we also have mystery. Okay, questions, unsolved question in terms of the chemistry. For example, decolorization is possible, but we still don't know the exact chemical mechanism. Okay, and also delipidation is possible by using some potent chemical, but we still don't know the exact underlying mechanisms. And then at that time, decalcification is a little bit difficult. And also, RA matching, refractive indices matching is also the issues. Okay, and then sometimes we observe the expansion of the tissue, which might be able to contribute to the tissue clearing, but we still don't know the exact mechanism. Okay, there are a lot of questions. Especially for the chemistry. Okay, so therefore we decided to perform the chemical profiling approach to analyze and understand the chemical property for the tissue clearing. Okay, so and then Tatsuya in our lab performed a heroic effort, and then he developed 5 different assays to ask, what is the chemical nature which can contribute to the delipidation or decalcification or decalcification, I mean bone clearing, and also expansion and RI matching. Okay, and then we found a couple of best performing chemical by using this screen, and also we perform the profiling of these chemicals to understand the chemical principle. And then we found some functional structures of the chemical seems to be, contributing to that each properties underlying tissue clearing. Okay, so here's an example. So when you combine the delipidation and RA matching, and apply them to the kidney, you can perform the whole kidney profiling. Okay, we just stain the kidney by using nuclear staining dye, and then as you can see here, you can extract the every single glomeruli existing in the mice kidney. In this case, we found 12,662 glomeruli existing in the mice kidney. Okay, and also that was a kind of a normal healthy tissue, but you can apply this to the pathologies, like cancer pathologies. So we put the we try to look at the cancer metastasis, which spread all over the mice bodies, which is highlighted in the red fluorescence. And then you can see here the some metastasized cancer cells exist in some organ in the mice. Okay. And then that was a mice application, but we can also apply this technique to the human diagnosis in collaboration with the pathologist in Osaka University, Professor Kojima. And then he is interested in the cancer metastasis to the, lymph node. And then because, diagnosis, detection of the metastasized cancer cell in lymph node is critical for the, decision of the treatment. And then by using tissue clearing technique, you can increase the sensitivity of the detection of the cancer cells. And then you can also apply this technique, not only rodent, but also to the primate. So in this case, we can apply this technique to the entire marmoset to look at the entire body of the marmoset. Okay. And also you can apply not only non-primate, but also humans tissues, including heart, liver, spleen, kidney, brain, and lung. And actually, we find out the very potent chemical which can apply to the human. So therefore, you can clear these organs. And then we are currently thinking to apply these chemical not only a block of the tissue, but also to the entire tissues, okay, of the human. But we are lacking the intact entire organs of the human. So therefore, if you are interested in becoming a transparent human being, please let me know. Please express your interest in Q&A sessions. We need volunteers. Okay. So after these chemical screening, this technique is now on the market. So you can buy these compound or chemical cocktail from the Tokyo Chemical Industry and then TCI. They are recently considering to change their name from Tokyo Chemical Industry to the tissue clearing industries. Okay, because these product is getting very popular. Okay, all right. But after these products get on the market, we encounter the unexpected applications. Okay, especially to the non-model organism. Here is an example. So Professor Konno and Okazaki independently apply our technique and they slightly modify our technique and to the pill bags. And then they created the transparent pill bag. Okay, and also they also succeeded to create the transparent crab as shown here. And then they try to look at the inside inner structures of the pill bag and also crab. Okay, and also here is another interesting story. Japanese high school student get interest in our technique, and then he decided to apply our technique to his favorite insect, like a drone beetles or a horn beetle and the stuck beetles. And then all of them become very transparent. So melanin, or color was decolorized by using hyperperoxide. And then after that, they treated these insects by using our tissue clearing reagent, Cubic. and then succeeded to make a, how to say, transparent insect. And then actually he got the award based on these, how to say, result. And also he recently wrote a paper, even though he was just a high school student. That was amazing, okay. Okay, and how about the bone? Okay, so in 2018, we succeeded to clear the bone to look at the bone metastasis of the cancers. And then we found not only chelation of the calcium by using EDTA, which is already known in previous pioneering work, but also protonation of the counter ion, phosphate, seems to be important, which is mediated by the imidazoles. Okay. And how about the decolorization of the heme? Okay. So as I said, interaction between heme and hemoglobin is important, seems to be important. And the interference between these interactions might be a key. But that was a pure hypothesis based on the structure of the amino alcohol, okay? But after 1,600 chemical screening, we found a very potent chemical, which turned out to be a 1-methyl-imidazole. And then interestingly, these structures seems to be very similar to the histidine structures, which is important for the interaction with the heme, which support our hypothesis in which interaction between heme and the hemoglobin seems to be interfered by the decolorization chemicals. Okay. Now, how about the expansions? Okay, so two graduate students in our lab get interested in the tiny structure like a synapses, and then they try to look at every single synapses existing in the mice brain. But we have issues of the resolutions. So therefore, they decided to expand the brain 10 times in volume, 2.3 times in length, as Edo Boyden nicely showed in 2015. So in Edo Boyden's work, he used exogenous polymers. But in our technique, we don't use exogenous polymers. We just put the mice brain into the chemical cocktail and then waited a couple of days. And eventually, adult mice brain slowly, gradually expand. And then by using identified chemical like imidazole and antipyrines. So once it expanded, we developed the customized light sheet microscopy to look at the structures. Okay, and then Tomo built his own microscopy, light sheet microscopy, equipped with a very good lens from Olympus. NA was 0.6 and the working distance 8 millimeters, 10 times magnification. And then he tried to observe the 1.3 million images to cover the entire mice brain, okay? And then turned out to be something like this. So tiled image is something like this, okay? And then we try to identify the hopefully synapses, but at the beginning we tried to focusing on the just nuclei, and then we found 100 million cells existing in the adult mice brain. Okay, we don't know, this 8 is exactly correct or not, but we are confident that it is larger than German population. Probably and definitely it's smaller than entire Indian population. But now we can deal with the 100 million scale of the cells by using single experiment, just single experiment. Okay. But interestingly, during this process, we found, just putting the tissue into the chemical cocktail, it gradually increase the volumes, which remind me of the very famous, hypothesis proposed by the Toyoichi Tanaka, who was a MIT professor, and then he had proposed our tissue is just the hydrogels. And then his hypothesis based on the purified, or DNA, purified protein and the purified sugars, polymers. And then he proposed our tissue is maybe, or hydrogels. And then we tried to test this hypothesis in more rigorous way, okay? Because it's gradually expand. And then therefore, we try to test by changing the environment, changing the pH, changing the osmolarity, and then changing the hydration. And then we found our tissues gradually expand and gradually shrink, gradually expand and gradually shrink, meaning our tissue act exactly like a hydrogels, okay? So, if so, our tissue can be described, tissue light property can be described by the very simple equations, like a Lorentz equation, which can characterize refractive indices of the gels, okay? So, monomer, unit refractive indices and the monomer volume determine the composite refractive indices. And interestingly, delipidation, decalcification have an influence on the unit refractive indices. The expansion of the volume, of course, have an influence on the volume of the unit. And then resulting refractive indices should be matched with the refractive indices of the water. Okay, so by that, we can reduce light scattering, okay? And interestingly, each process can be achieved by the specific chemical functional group, okay? And also, in addition to the light scattering, we can reduce the light absorption by removing, eluting the pigment by decolorizations, okay? So if you're interested in that, please check our recent review paper, okay? So this is something like very early stage of the chemical principle, but we gradually getting such kind of a chemical principle in this field. So therefore, we feel it's high time to move forward from the basics to the applications. For example, we now have information of every single cell in the particular organ, like adult mouse brain. So therefore, we can, we may be able to construct some kind of atlas, single cell-based atlas of our brain. Okay. So actually, we constructed such kind of atlas. Okay. So atlas made of dot, okay? And then by using this atlas, we can map the many information onto the atlas. It's like a genome information, okay? Genome is a single nucleotide-based resolution, information that our inheriting, or substances, right? And then single cell atlas is single cell resolution, representation of the organs, okay? And then you can map cell type information onto the atlas, and the activity information onto the atlas, and the connectivity information onto the atlas, okay? So for that purpose, we also developed the tissue staining technique, okay? So after tissue clearing, penetration through the tissue is, getting increased, accelerated. So therefore, we try to test many buffers, and then we finally identify the right condition to accelerate the penetration of the antibodies. So therefore, we now, perform the whole brain immunostaining by using a bunch of different antibodies. Okay. Here is some example. And then this technique is published in Nature Communication this year, and also commercially available from collaboration between new venture company Cubic Stars and TCI. And also after we map these information onto the atlas, we can analyze the data. For example, if you identify the immediate early gene expression, like a CFOS protein expression all over the brain, you want to analyze what kind of a brain region are highly activated and what kind of a brain region are highly suppressed after administrating drug or changing the experimental conditions. Okay. So by using this, software, cloud-based software, we can map such kind of information onto the atlas and then compare the different brain sample on the cloud. Okay. So here is an example. After analyzing these immunostaining data by using Cubic Cloud service. For example, if you look at the PV data, this is a para-albumin data and the somatostatin neurons, which turn out to be the 0.5% of the entire brain cells. Chat neurons and TH neuron is 0.07% and 0.08%. And the CFOS CFOS expression is depend on the drug administration. For example, after NMDA receptor inhibitors, 4 times more cells are activated after drug administrations. And also you can use this technique to the analysis of the cell circuit by using Davis virus technique. And then actually, these information can be mapped onto the 100 million atlas cells. Okay. And then just recently, these work is also commercially available. Okay. Okay. So in the last couple of minutes, I just touch upon the future directions. Okay. So one direction is becoming faster. Okay. So by using previous microscopy, it was a little bit slow, but we can now accelerate the imaging time down to the 5 minutes for 10 gigabyte and 4 hours for 10 terabyte. Okay. And then another direction is becoming, getting smaller. And as I said, we are interested in the whole brain synapses profiling. And then we are now combining expansion microscopy technique with the tissue clearing together with the high resolution microscopy. And then we try to get that data, like a 1 petabyte data for the one entire mice brain. And so therefore we call this project as a Petascope project to look at the synapses, whole brain-wide synapses. And the final direction is getting bigger. As I said, now we can, analyze the entire infant body cell profiling. And as I said, more than 400 million cells can be detected. And then also we can apply this technique, not only mice infant, but also marmoset infant. And then as you can see here, entire bone and the entire organ is completely cleared. melanin is still issues, but we'd like to figure out. And then you can apply this technique to the now non-primate, a non-human primate. Okay. And then here is a adult marmoset brain, and then we have a project to get the single cell resolution atlas of the entire marmoset brain. And also, we recently succeeded to stain the hemisphere of the marmoset by using tissue staining technique. Okay, and finally, so one of the biggest challenge in this field is to make a entire human brain single cell resolution atlas, which is hard, but we'd like to achieve in near futures. Okay, so today I talked about the cubic technique and the application of these technique to non-human primate and humans. Okay, so if you're interested in becoming one of the first transparent humans, please let me know. So thank you for your attentions.
Thank you very much, Dr. Hiroki. It was really nice and fascinating talk. I believe that our audience is looking forward to see a human brain mapping, and we would like to see your application coming into the real, clinical setting where the patients of, maybe any cancer or neurodegenerative diseases get benefited, and we can predict some of the disease condition by using your technique. It's really fascinating, and we really have a different flavor than what we had so far in this conference. So we have a couple of questions from the audience if you would like to take one or two questions.
Okay, yep.
So the first question is from Asmita Dasgupta. She's asking, does this mean that changing the height of illumination change the layer one is viewing in the tissue?
Does it mean that changing the height of the illumination change the layer one is viewing it? Yes, yes. So so you can illuminate from the side, and then if you change the height, you can illuminate other layers, and then you can repeat these things to cover the entire organs one by one. So usually we can cover the so in the 10x microscopy, we try to look at the 3,000 images for one particular lesion, and then we repeat it 400 times, and then eventually we got the 1.3 million images to cover the entire brain. Yes.
Okay, so we have a next question. If you can see from chat box, please.
Okay, yeah, is the tissue expansion isotropic? Yes, so we sometimes, for example, olfactory bulb is a little bit deformed, but we check the isotropic feature of the expansion during the process. And then local and the mesoscopic feature is isotropic, yes.
Okay, there's one last question. Is there still in the line? If you can just quickly recommend.
Okay, so confocal microscopy. Okay, so light sheet microscopy is a little bit, quite faster than confocal microscopy. So therefore, because you can illuminate the one 2D dimensional image, and then you can immediately get the one 2-dimensional image. Okay. And then CMOS sensor is highly, very speedy. So therefore, you can fully, utilize advancement of the CMOS sensor. So usually we can get the 50 frame per second. So you can get the 50 image within 1 second. So it's quite fast.
And yeah, we have another question.
Yes. Could you pick up something?
The last one, the fascinating, does this eliminate the autofluorescence by
Oh, I see. Yes. Okay. So, in the human, we still have an issue in the autofluorescence, especially probably from the lipofuscin. So in mice, we don't have such issues, but in human, we have issues. So therefore, we try to, reduce such autofluorescence by, treating the, some chemical to reduce the autofluorescence. That's still ongoing work. Yes.
Anyway, thank you, audience, and thank you to the Dr. Hiroki. Such a nice and wonderful talk, and we wish if we can meet sometime in the future.
Yeah.
Thank you, everyone, for joining us, and thank you very much for giving time for the IAN. Thank you very much.
Thank you. Thank you so much.
Hello.
Thank you.
So, Professor Hiro, I can see that everybody has once again enjoyed your lecture. And
Thank you.
I'm really very happy that you accepted and you adjusted to the time, which was though not suitable to you.
No, it's okay. Yeah, thank you. Thank you so much.
Thank you
Yeah.
so much. Thank
I'd
you.
like to visit, India again.
Yes.
Yeah.
So always, as the bill says, always there's a next time. So I will
yeah.
invite you certainly to Hyderabad.
Okay, yeah, okay.
You can enjoy Hyderabad delicacies that time, and we
Okay.
can always through our mails and can interact more. Thank you
Okay,
so
yes,
much. Thank
yes.
you. Thank you.
Thank you, everyone. So the oral session 1 will start on the same link in about 10 minutes. Good evening. If the chairpersons are here, you can start the session, please. Thank you.
So very good evening. So can we start the session?
Hello, Professor Pant? Dr. A.V. Pant?
Yeah, you're getting my voice?
Yeah, I can hear you. Sir, I think you are the chairperson for the session 3.
So I know there may be
Hello?
still more ideas
This is,
and queries.
this is not
This
session
is
3.
this is oral session 1, sir. I'll just send you the link in the chat box.
Redirect our
I will leave and I will disconnect.
thinking process in the current scenario. And so with today's discussion so far, it seems
If Professor S.K. Trigun and Professor Rajat Sandhir are here, you can start the session.
Yeah, we are here.
Okay, thank you so
So
much, sir.
I'll request Dr. Trigun to start the proceedings for the session.
Okay, thank you, Dr. Sandhir. Am I audible?
Yes, you are
Please
audible.
respond.
You are audible. Yeah.
Okay. Oh, okay, sure. So, good evening to everyone, and this is my proud privilege to first start this session. This is oral session 1. Before that, we have one important lecture by Dr. Patel as invited talk. So, if you permit, Dr. Sandhir, instead of introducing ourselves,
Yeah, I think that makes sense. That makes perfect sense.
Yeah, that otherwise we know each other. So for audience as well, I think we are more visible in IAN. So with this, I have privilege to introduce the invited talk speaker, Dr. Anant B. Patel, whose affiliation as on now is his senior principal scientist in CSIR Center for Cellular and Molecular Biology, Hyderabad. And his another affiliation is with Academy of Scientific and Innovative Research, Ghaziabad. His research interest is in neurotransmitter energetics in depression and so many other diseases, including Alzheimer's, Parkinson's. And he has, for his nice work and contribution in this area, he has been recognized in very many ways. To name a few, he has been an associate editor of Frontiers in Aging Neuroscience. He is member program committee Brain 2. This is a biennial conference organized by the International Society for Cerebral Blood Flow. He is a recipient of Professor A. Subramaniam 60th Birthday Lecture Award 2070. This is the National Magnetic Resonance Society of India. So with this brief introduction, I would now request Dr. Anant B. Patel to kindly present his talk.
Okay. So I'm trying to share my slides. Just to give me a minute.
Sure. You take your time.
Should we log in again?
By that system, you were able to share from that time. It is asking some login preferences or the text of sharing.
Professor Anant P. Patel, are you here?
Together and importance of the
I think Professor Anant Patel is facing some connectivity issues. Let's just give him some time to join back.
Let us give some time to Dr. Patel.
In the meantime, Dr. Trigon, how much time do we have for oral presentations? Can you announce that for the participants?
Actually, total time for 5 lectures and plus one, I think 30 minutes we can, is invited talk. So I think we can go for 30 minutes. And then in remaining 1 hour, 15 minutes, I think we have 5 talks, oral presentation. So 15 minutes each, I think it will be over.
Yeah, 15 minutes each would be good. Yeah.
For oral, let us give 15 minutes. So it depends upon
somebody
how
call
much time
Dr.
Patel.
Yeah, can somebody call Dr. Patel to know whether, if he allows, we can have one oral presentation in the meantime to say, in the interest of time.
Yeah, this is the right idea. It's a good idea. If he has some
Please go ahead. Please go ahead with that first oral presentation.
Okay. Isn't that first one presenter, Anil Annamadi is ready?
Yes, sir. I'm here.
Okay, that's good. That's good, Anil. So you will have maximum 15 minutes.
Yep.
So please go ahead with your presentation.
Okay, I will try to share my screen. Can you see my presentation, sir?
Yeah, we can see your presentation.
Yeah, it is visible. You may go ahead.
Okay. So first of all, good day to everybody from different parts of the world. And also I would like to congratulate the organizers for organizing this meeting in such a wonderful way. And also thank you for selecting my abstract for the oral presentation. So today I'm going to talk about presynaptic protein BUS-OON and its role in GABAergic interneurons in mediating learning and memory. Due to time constraints, I will only talk about the behavioral changes. Okay, so you see here the synaptic button, which contains the presynaptic and the postsynaptic compartments. The active zone is a part of presynaptic compartment where the synaptic vesicles fuse, dock to the membrane and release the neurotransmitter into a synaptic cleft. If you look into the 3D architecture of this active zone, it's a quite dense meshwork of proteins, and BUS-OON is one of these active zone proteins, together with piccolo, ring binding proteins, RIMs, and so on. So if you look into the BUS-OON, which is quite a big protein with 420 kilodaltons molecular weight, and it has a lot of interaction partners, as you can see here, through which it mediates different functions, important functions at the presynaptic compartment, which I try to draw in a representative way. To list out a few of important functions, it is involved in localizing the PQ type of calcium channels close to the synaptic vesicle, which is docked through interaction with ring binding proteins. And it is all involved in homeostatic synaptic plasticity. So through interaction with CTBP1, which is a nuclear transcriptional repressor, which shuttles between nuclear compartment and the synaptic compartment, BUS-OON involved in regulating the synaptic nuclear communication. So recently, we are studying a lot about BUS-OON function in regulating the protein turnover integrity at the presynaptic compartment. We are able to show that through different collaboration partners, BUS-OON is involved in autophagy and the proteasomal degradative pathways at the presynapse. BUS-OON is also shown to be regulated in cortex-dependent learning paradigm, implicating its role in learning and memory. If you look into the life without BUS-OON, there were two constitutive knockouts or the total knockouts generated in our lab. However, both of them.
Dr. Anil, would it be possible to be louder? Bit?
Yeah, sir.
Yeah, please.
So these both the constitutive knockouts display severe epileptic seizures, and also they display sensory impairments like vision and auditory impairments. Hence, these total knockouts are not suitable for behavioral assessment. Thus, what we did, we started generating conditional knockout allele by inserting LOXP sites around exon 2 of the BUS-OON gene, which contains 12 exons. Upon Cre recombination, premature stop codon comes and then silence the gene. Through using different Cre driver lines, for example, EMX-Cre, we generated conditional knockout of BUS-OON in forebrain excited neurons, which is B2E. And then through DLX 5-6 Cre driver line, we generated a conditional knockout of BUS-OON in forebrain inhibitor neurons to address the specific behaviorally relevant effects of BUS-OON deficiency in animals. What we published a couple of years back, that I quickly summarize this. When BUS-OON is not there in excitatory presynapses, which you can see here in the V-glut positive presynapses, leads to impairment in proper maturation of dentate gyrus. These dentate gyrus, or the medial perforant path to dentate gyrus, has the juvenile state, which at the end affects the DG-dependent learning, what we show in pattern separation and background contextual fear conditioning. So this part I also published, I also presented in IAN meeting in Varanasi 2 years back. Okay, now after this, we move ahead and then started characterizing the second conditional knockout of BUS-OON in forebrain inhibitor neurons. What I did here, immunohistochemical analysis, you can see the VGAT, which is a marker for GABAergic interneurons, presynapses, lack BUS-OON expression, whereas the V-glut, the excitatory presynaptic compartment still express BUS-OON, indicating the specificity of this knockout. Okay, so the knockouts are specific, and then I continued with the behavioral screening of these animals. So I did a home case analysis to check for the circadian activity, which is quite normal. And then I look into the activity in novel environment by placing the animal into open field, where you can also measure the anxiety behavior by the time they spent in different compartments like a center or corner. Here, I would like to show you a few videos. So this is a wild type condition when I place the animal into the open field, and you can see that it's quite okay and exploring, and also thinking that whether to go around, but then it decided to explore the whole compartment, going to different parts of the open field. And then I placed the knockout animal into the open field. As you can see that they are quite hyperactive in this novel environment. And then they think that, okay, should I go to the center or not? So there is like come and go, but then at the end they confine themselves to the corners or the rims, but not to the center area, which is also, you can see here in the quantifications, the total distance they cover in the open field, which are quite high in the knockouts, and also the time they spend in the center arena. You can also see this in representative heat map images. So further, I also look into the anxiety phenotype in two more paradigms, like elevated plasmas, where you can see the time they spend in the open arm, quite less in the knockouts. And also in the light dark test, you can measure the activity in light compartment to see the anxiety phenotype. So with this, it is quite strong phenotype, the anxiety phenotype in these B2I knockout animals. Then I moved on to next parameter, social behavior. Here, first I look into the in a three-chambered test, sociability, where there are two opaque cylinders where you can put a mouse in one cylinder and then the object in another cylinder and see the test mouse, the preference of the test mouse in this sociability test. During the social memory or recognition, the mouse which is they already encountered will be a familiar one, and then I replace the object with a novel. So the time they spend with the novel mouse is an indication for the social memory or recognition. As you can see in the sociability test, both the groups, they prefer the mouse, not the object, which is quite good. You can also see this in heat map images. When it comes to the social memory, the wild type, without any surprises, they prefer the novel mouse compared to the familiar. However, the knockouts have problems in identifying or recognizing the familiar and the novel counterpart, which is also seen here, indicating they have problems with the social recognition or memory. So our collaborators, they performed another test to check the social interactions. So what they did, they placed the urine of the female estrus mouse into a chamber and see how the test mouse behave. You can see that the wild type situation, they are more close to the urine, whereas the knockouts, they ran away from the urine of the female mouse. You can also see this in the quantifications. Next, they provided the female mouse into the chamber. Here, I want to show you the video. So this is the wild type situation. So there is a female mouse which is small, and then we place the wild type male, and you can see that the male mouse do like genital sniffing mouse, nose sniffing, body sniffing, and so it interacts with the females. It actually also make some calls. I'm not sure whether you can hear any calls from this video. Yeah, anyhow. So next, they place the knockout male, and you can see it confined again to the corners, which is having like no interest at all. It's like by accident one time they did it, but you see that the male knockouts, they don't do any sniffings or something, but whereas the female actually is following the male here, and the male is running away from the female. So which is you can see here in the quantifications, like different social interactions, and also I think you did not hear any ultrasonic vocalization from the videos, but the knockouts also did not make any USVs. Only the wild type, they make these calls. So together, the B2I knockout animals display also the social behavioral deficits. Further, I tested nest building in these animals, which is a parameter for general health in the rodents. So what I did, I provided a cotton nestlet into the home case, and we give the rankings as you see here. If they build a nice nest with a burrow and the walls, they get the score 5. If not, they get so depend on how they build the nest. You can see in the wild type situation, they are quite good. They build a nice nest. However, the knockouts, they did not build any nest. So the maximum score they reach is less than 2 in the case of knockouts. Altogether, what these results suggest that Bassoon deficiency in the GABAergic interneurons results in very pronounced emotional and social behavioral changes, which are quite reminiscent to autism spectrum disorders. It's also quite interesting because there are several autism genetic models of autism or immunological models of autism, where you see any one of the parameters like anxiety or a social behavioral deficits or nest building or something like this. But surprisingly, in our model, we see all the parameters one can see in different autism models reported till date. So this is quite interesting and can be used as a general model to study network activities or to test novel therapeutic drugs using this model. Finally, to give a brief outlook, recently I received independent grant to conduct my postdoctoral research. So here I'm trying to look Bassoon expression in different autism spectrum disorder models to see is there any causal link between Bassoon and the autism. So we have in our in Magdeburg in Germany, we have different models available: SHANK2, SHANK3, Fragile X, MECP2, which are genetic models or acceptable models for autism. And then I also started generating another conditional knockout of Bassoon in parvalbumin interneurons, because these are the subtype of GABAergic interneurons which are more prone to changes in neurodevelopmental or neuropsychiatric conditions. Finally, I also received another pilot grant together with another colleague. And here we are looking into proteome changes in different Bassoon mutants to see if there are genes or proteins which are related to these disorders or regulating in Bassoon mutants. And we can then delineate the signaling cascades underlying these phenotype, what we observed. With this, I would like to thank my supervisors, like current supervisor, Professor Oliver Stark, and then my previous supervisor, Professor Eckhard Gundelfinger, who actually isolated or identified the Bassoon and Piccolo proteins from the presynaptic compartment, and the close collaborators, who's Gursel, who did the electrophysiology, and then Carolina, who was involved in vitro cell culture data, and the collaborators and the funding agencies. This I would like to thank you all.
Okay, thank you, Dr. Anil, for finishing your talk in time. We have a chance for 1 or 2 questions. If anybody is interested, they may address the question. In the meantime, this Bassoon, your model, how much it is close to the real autism condition? I mean, any neurobehavioral parameter which you can claim that this can be a replication of or reproduction of a real autism condition? Because this is the main problem we all are struggling with, that rat doesn't say anything and we interpret the way we interpret. So since you are claiming, and therefore this makes another important issue. So can you comment on this?
That's quite good question, sir. As you said, they don't speak, but what we can see from our model or compare with other models is the behavioral phenotype, what we see, like several behavioral changes or the parameters which are disturbed in this model. And also, I thought I don't have so much time, so I did not show anything. Like I already did some preliminary experiments in the SHANT 3, where I see almost half of reduction of bassoon. And also from the proteomics, which we just received like a few days back, I started digging into the genes or proteins which are related to autism. There are actually several of candidates which are regulated in this model. So yeah, we still don't know whether it's really acceptable, but from the literature and also with the different mouse models which are available in our lab, we think that this is more generalized model.
Okay, that's good. I mean, we all are struggling with the same concept, and therefore this is not a question only for you. For all of us, those who are working in neurochemistry, molecular neurobiology, and then correlating with neurobehavior. So anyway, thank you so much for your nice presentation.
There's a question, Dr. Trigun, for him from one of the participants, Ray. He's asking whether these findings would translate to depressive disorders also.
Okay. Meanwhile, can you unshare your slide so that I can try sharing my slide?
Yeah.
So depression, I'm not sure actually with the another model we have, like the excitatory specific one. And also we also have a dopaminergic specific.
You can stop sharing your slide so that Dr. Patel can.
Okay.
And we can keep discussing in the meantime.
Yeah.
Okay. Anil, I also have one question for you. Like you talk about GABAergic neurons. Does this bassoon also have a role in other kind of exocytosis also? And are there studies linking bassoon mutations with some kind of neurodegenerative disorders?
So there has been one mutation identified recently from Japan in the progressive supranuclear palsy-like syndrome, tauopathy, where they identified both familial and also sporadic mutations in bassoon. And also in intellectual disability patients, there are mutations identified from Jordan, which is published, and also from like in our collaborators lab, which is still not published. Yeah, so far these are the mutations, but the mutations which are also linked with the epilepsy, like Klafnov syndrome, syndrome, which is early childhood epilepsy. And bassoon is also, mutations of bassoon is also seen in one of the deafness syndrome. So actually these are, so we still don't know if it is going in the direction of neurodegeneration or neurodevelopmental or neuropsychiatry. Yeah, we
Thank
are.
you.
Thank you, Anil, for nice presentation. I enjoyed listening to your talk. Thank you.
Okay. So now I think Dr. Patel is ready.
Dr. Patel is ready, so yeah, we can.
Dr. Patel, please go ahead.
So I'm very sorry for this last minute some technical problem.
Yeah, technology can do this to everybody, so doesn't matter.
I think people can see my slide.
Yeah, all of us can see you
and hear me.
Yeah. Go ahead.
Professor Trigun, am I audible there?
Yeah, sure. Go ahead.
Your voice is perfect.
Perfectly all right. Go ahead.
So, is coming.
You have some problem? Slide sharing?
Okay, so at the outset, I'd like to thank Professor Trigun for saying a kind word for me. And before I start my lecture, I will start by thanking Professor Anita Jagota, and Professor Shashi Bala Singh for inviting me for this presentation. My lab is interested in studying brain energy metabolism in different neurological disorders like Parkinson's disease, Alzheimer's disease, amyotrophic lateral sclerosis, and psychiatric disorders like depression. But today, for the sake of time, I will be considering only for depression-related study. Okay, so as we know that information in brain is transferred by means of neurotransmitter. Here I'm showing you synapse where lot of neurotransmitters are packaged into the vesicles. And when the action potential reaches at the terminal, there is a release of neurotransmitter in the synaptic cleft, and these neurotransmitter binds to the receptor present on the postsynaptic neuron. And then action potential propagate to the next cell. Neurotransmitters are ultimately cleared by astrocytes where it is converted to glutamine and then returned back to the neurons. And this cycle we call neurotransmitter cycle. In physics term, you can see that this is a kind of work done by the neurons. And to support this work, neurons require a lot of energy. And energy requirement of the brain is very, very high. That will be seen from the fact that our brain is only 2% of the body weight, but it contributes around 20% of that energy required by the body. And most of this energy is supported by oxidation of the glucose in the brain. So by NMR spectroscopy, we study neurotransmitter cycling flux and neuronal glucose oxidation in the brain. So for this purpose, we use tracer approach where non-radioactive 13C levels glucose is administered into the blood. And then it is transported to the brain, and through TC cycle, label is transferred to the different TC cycle intermediate, and ultimately it goes to the neurotransmitter glutamate. Then this glutamate fired, it binds to the postsynaptic neurons and then taken by the astrocyte. So here idea is that you can follow the labeling of glutamate from glucose to quantify the TC cycle flux. And that will tell us glucose oxidation because half of the TC cycle will be glucose oxidation. And in the same experiment, we can monitor labeling of glutamine to measure the neurotransmitter cycling or glutamate-glutamine cycling. Now, here is one example. This is an MRI spectrum recorded from the human brain without infusing glucose, that lower panel. You don't see much signal because 13C enrichment is only 1%. But if you infuse glucose, 13C level glucose, and then record spectrum, you see that different signal coming from neurotransmitters, glutamate, GABA, and then aspartate and all those. So using this approach, neurotransmitter cycling and neuronal glucose oxidation has been evaluated with varying level of brain activity, starting from human to rat or mice. And you can see that with increase of the brain activity, neurotransmitter cycling flux increases, and at the same time, neuronal glucose oxidation also increases. The most important thing is that these two processes increases linearly and with slope 1:1. That tells that neurotransmitter cycling is stoichiometrically coupled with neuronal glucose oxidation. That means to cycle one molecule of neurotransmitter, neurons require some energy, and that energy comes by oxidation of the glucose in the neuron. So therefore, now we can monitor brain activity, which was earlier done by electrophysiology and neurophysiology we called, by indirectly monitoring that neuronal glucose oxidation. So now coming to the depression, it's the most common neuropsychiatric disorder. And as per WHO recent reports, around 265 million people are affected with depression. And it is the leading cause of disability worldwide. If you look at the contribution of depression to the different neurological disorders, globally it contributes almost 30%. In India, it is in fact 40%. Most of the studies related with depression earlier was carried out following that different monoamines. But recently now there is more interest in the glutamate and GABA, which are the major excitatory and inhibitory neurotransmitters. It accounts around 90% of the total neurotransmitter present into the brain and synapse. Dysfunction in these pathways has been implicated in many neurological and psychiatric disorders. And our prefrontal cortex is associated with decision-making, learning, and reward pathway. And recent MRI studies has shown that there is atrophy in PFC of depressed subject. So therefore, our focus will be on the prefrontal cortex. Now we use a chronic unpredictable mild stress model of depression, where that a mouse is subjected to unpredictable true stress at a day for almost 3 weeks. These stresses are circular motion, cold stress, resistant stress, or tail suspension. And at the end of this stress, we want to see whether the animal is showing any symptom of depression, because the animal cannot tell. So we have to look at some behavioral measure. And here we are looking at the behavioral measure is sucrose preference, which is an anhedonia for the animal or human. So you see that control animal was taking around 90% sucrose, but after subjecting to that stress, now sucrose preference is decreased. And another measure is immobility. In under forced swim test, we looked at how long the animal is immobile. So immobility time is increased significantly in that animal subjected to the cones. Under this condition, we wanted to study what is happening to the neuronal function in these animals. And our interest is prefrontal cortex. So for that purpose, we are using another substrate, acetate, which is labeled at carbon 2 position. It is given into the blood, and then it is after transport into that astrocytes, it is metabolized there, and then first incorporate labels into the glutamine carbon 4. And then during synaptic transmission, label goes to that either glutamatergic neurons or GABAergic neurons, and therefore labels glutamate at carbon 4 or GABA carbon 2. So if I can account all these labels from a start infusion of acetate, that will tell us about acetate oxidation. And because acetate is selectively transported into the astrocytes, we can actually measure that astroglial metabolic activity using this approach. So here is one example. After 15 minutes of infusion of acetate, we have recorded an MRI spectrum from the prefrontal cortex. And then I want to put your attention on the lower panel, where we are seeing that a leveling of glutamine and glutamate. So this has been quantified and put here. And you can see that amount of label incorporated into the glutamate, GABA, and glutamine is almost reduced by 20%. And then once you calculate that cerebral metabolic rate of acetate oxidation using this approach, we found that almost a 25% reduction in that acetate oxidation in the brain. And because acetate oxidation is occurring into the astrocytes, we see that the astroglial activity is decreased. So next, we wanted to understand what happens to the neurons in the depressed mice. For that purpose, we have infused 1,6-13-C-glucose. And as I mentioned earlier, after transport into that brain, it is converted to that pyruvate, and the label goes from the carbon 1 of the glucose to the pyruvate 3. And then that is converted to the acetyl-CoA by pyruvate dehydrogenase pathway, and then further oxidized by the TCA cycle of the either glutamatergic neurons or GABAergic neurons. So during that process, it labels TCA cycle intermediate, and ultimately label goes into the glutamate carbon 4 position or GABA carbon 2 position. If label travels further into the TCA cycle, then it goes to the carbon 2 and 3 of the glutamate or 3 and 4 of the GABA. And then these label neurotransmitters ultimately fired on the postsynaptic neurons and taken by the astrocytes and label go to that glutamine carbon 4. So by following the labeling kinetics of these amino acids, we can quantify the TCA cycle flux of either glutamatergic neurons or GABAergic neurons or neurotransmitter cycling. So just to give you an example, here we have infused glucose to the animal at different times, 10 minutes, 30 minutes, or 60 minutes. And you can see in the 10 minutes, we see only signal coming from the glutamate carbon 4 that increases with time. But if you later time, you see that there is signal coming from the glutamine carbon 4 or glutamate carbon 4. And similar as signal was obtained in the CUMS mice, but there was some delay. And that has been quantified and then plotted. And then here is the kinetic of labeling of that amino acid glutamate carbon 4 in the control brain, the solid line. And then dotted line is in the CUMS mice. And you can see that flow is slower in the CUMS mice. Similarly, glutamate carbon 3 is shown in the lower panel. And then GABA 2 or 3 is also shown. And this has been analyzed using a metabolic model. And then whatever the result outcome for the metabolic rate is presented here. So you see that level glucose oxidation in that entire brain, irrespective of whether it is glutamatergic neurons or GABAergic neurons or astrocytes, is decreased significantly in the CUMS mice. And generally, when you are doing PET autoradiography and all those things, we are looking at total glucose consumption, and that is, this data is consistent with that one. But our interest was that what happened to that excitatory neuron or inhibitory neurons in the CUMS mice. So if you look at excitatory metabolic activity for the CUMS mice was decreased almost 20 to 30%. GABAergic related inhibitory activity was also reduced, but not to that extent of that glutamatergic activity. If you look at the glutamate-glutamine cycling, that was decreased, and GABA-glutamine cycling was also decreased. So this was that observation we made in that CUMS mice. We also used that another model, which is chronic social defeat mouse model of depression. In this case, a test mouse, C57BL6, is subjected to social defeat for 5 minutes. We're using a very dominant CD1 mice for 5 minutes. And after that, these two mice are separated with a transparent barrier of glass. But this mouse will be seeing the presence of the dominant mice. And then next day, again, we repeat the defeat with another mice, and this process continued for 10 days. After 10 days, again, we looked at the behavioral phenotype, sucrose preference, and social interaction. So we see that around 2/3 mice shows reduced sucrose preference, and those mice we categorize into the depressed mice, but 1/3 did not show any reduction in the sucrose preference. So that we call resilient. Similarly, if you look at the social interaction also, around 2/3 mice were showing that a reduction in the social interaction, but 1/3 did not show. Then we characterize the energy metabolism in this brain by infusing 13C-level glucose. And you can see that mice which are showing the depression-like phenotype, their glucose oxidation in glutamatergic neuron is almost 30% reduced. GABAergic pathway are also affected, but to a lesser degree. And then we looked at a gene associated with the glutamate and GABA pathway, and we found that excitatory amino acid transporter, which is responsible for the clearance of glutamate from the synapse, that expression was decreased in defeated mice. And another gene, which is responsible for the synthesis of the GABA, so GAT1, that level is also, expression is also decreased by almost 50%. So these are our lessons from that stress model of the depression. Recently, we have collaborated with Dr. Vidita Vaidya, and she has presented a part of that work. I just want to show you that data related with that metabolic measurement. And in this case, she has used a bigenic mouse model. And in these mice, GQ signaling pathway was activated by administration of clozapine N oxide, starting from postnatal 2 to postnatal 14 days. And then these mice were assessed after 3 months. And as she has shown earlier also, that these mice preferred less time in that central area, and INT is also reduced. And if you look at a light dark assay, in that case also that their entry into that time into the light area, entry into the light region is also decreased. And in post-stream test, they are showing actually more immobility. So then we studied again energy metabolism in these mice by infusing 13C glucose. And I want to draw your attention for only 2 things. Here, glutamate carbon 4 labeling, and then cerebral metabolic rate of glucose oxidation by GABAergic neurons. So unlike in a stress model, here we are seeing that there is increase in the neurometabolic activity in hippocampus. And when you go for do the same analysis for the cerebral cortex, there we find that in addition to the GABAergic activity, glutamatergic activity was also enhanced. So there was more changes in the cerebral cortex compared to the hippocampus. So these are our understanding about energy metabolism of excitatory and inhibitory pathways. Now we wanted to see whether we can somehow ameliorate that symptom of depression in these conditions. So we tried one formulation which is called Rasa Sindur. It is used in our ancient tradition that it is an organometallic based on the mercury and sulfur. And in our tradition, it is used for regenerating the rasana therapy. And it is believed that it promotes the long life with enhanced physical and mental strength. Moreover, in fly model, Lakotia's group has shown that in normal animals, it actually prevents the accumulation of heat shock proteins, suppresses apoptosis, and improves the physiology in the transgenic model of Drosophila, which are VS, Alzheimer's, and Huntington-like condition. In addition, in our rat model, also we have shown that it improved the brain energy metabolism as well as memory in AD condition. So we wanted to look at the impact of these formulation in social defeat model and of the incomes model of the depression. So here is the paradigm. First, we have actually done 2 experiments. In one experiment, first we have given this RS, and then followed by 3 weeks of the COMS, and then behavioral analysis, and then metabolic measurement. In another group, first we have given actually developed the depression, and then that is followed by intervention of the this RS for 4 weeks, and then again measurement of the behavior and neurometabolism. So here is the data for the pre-intervention with RS. Like before giving that stress, we are giving first RS, and you can see that like if you give the RS and give the stress, then we don't see any reduction in that sucrose preference, and we don't see any. We actually see that a reduction in that immobility time in the 4-stream test. And then we carried out metabolic analysis in 2 regions, prefrontal cortex and cerebral cortex. So again, if you look at the last arrow, last bar compared to the red one, we see the significant improvement in the brain energy metabolism of glutamatergic neurons and GABAergic neurons in the prefrontal cortex and cerebral cortex. And these changes are not uniform throughout the brain. We carried out measurement in the cerebellum, and cerebellum was not affected with the stress, and as a result of that, there was not much impact of that RS on that cerebellum. Then in the second experiment, where first we have given stress, and that was followed by that COMS, that RS. There again, we see that immobility of that animal was reduced almost maybe 2/3, although it is not very close to the control, but it was very different from that COMS mice, which was not given to that, which was just given CMC and not RS. And in these mice, again, we saw that there is an improvement in that brain energy metabolism associated with glutamatergic neurons and GABAergic neurons. So this is our understanding related with that Ayurvedic formulation. Now, I just want to show you some data related with looking at fast-acting antidepressant. So most of that antidepressant which are currently available, they are developed based on that monoamine targets and almost 3 to 4 decades ago. The major problem with these antidepressants are they are very slow acting, and then their low recovery rate, long remission time, and many times these are actually some people are not responding to this antidepressant. So now people are looking at alternative like antidepressant which can be like fast acting. In that case, people are looking at people are targeting the glutamate pathway, which I showed you that was actually affected in that COMS and social defeat model of depression. So in this case, there is some potential shown with the ketamine, which is an NMDA receptor antagonist. So ketamine is actually at a very high dose, it acts as an anesthetic. But it has been shown that at some anesthetic dose, a single dose of ketamine actually produces rapid and long-lasting antidepressant effect. In fact, ketamine is also effective in that people who are actually resistant to the different antidepressant. So we tested this one in social defeat model of the depression. And again, we have measured the behavioral thing and then neurometabolism. Here we carried out 3 behaviors: sucrose preference, which was very much almost 20% reduction in the social defeat mice, but with just one dose of ketamine, restored that sucrose preference to the similar to the control level. Similarly, the social interaction was also restored, and immobility time, which was very high into the social defeat animal, was decreased to the, actually in fact less than that normal animal, just by a single dose. And under this condition, when we carried out neurometabolism, you can see that here the leveling of glutamate in that control mice, and then in that defeated mice, amount of level incorporation from glucose is very less. And then when we treated these animals with ketamine, just 2 days dose, then leveling is now similar to that whatever like control condition. And that translated to that actually restoration of the energy metabolism of glutamatergic neurons and GABAergic neurons following ketamine intervention. We also looked at what happened to the astrocytes, and as I mentioned that astroglial activity was decreased, but when a ketamine just only one dose has restored the cerebral metabolic rate of state oxidation, almost similar level to the control. So just ketamine is just single dose is enough to improve the mood and that these social defeat animal. Similar experiment study was conducted in that human, and they have also shown that ketamine, just acute administration of ketamine, improved that leveling of that amino acids glutamine, but there is no change in the leveling of glutamine. So they propose that ketamine just improve the neurotransmitter cycling, but not neurometabolism, without affecting the neurometabolism. So although ketamine has shown the promise, but it has possessed the addictive properties and many other side effects like dissociative characteristics. So then to get rid of this problem, AstraZeneca has prepared a synthesized another molecule, which they call lansomine. This is also targeted to NMDA receptor, which is actually glutamate receptor, but its binding affinity to this NMDA is very, very low. So it's a low trapping NMDA receptor antagonist. And based on that animal study, it has been shown that it also has potential to that antidepressant characteristics, and also its side effect is lower compared to the ketamine. So we tested this drug in our CUMS model of depression, where first animal was given CUMS for 3 weeks, and then that was followed by lansomine intervention for 2 weeks, just only each week 3 doses, so total 6 doses. And then we carried out metabolic analysis. So here is that behavior data again. If you look at sucrose preference, which was much lower in the CUMS animal, but after giving that lansomine for 2 weeks, it is restored similar level to the control. And then immobility time was very high in that CUMS mice, and that is decreased to that almost control level after lansomine treatment. And then finally, we carried out neurometabolic analysis, looking at the glucose oxidation associated with glutamatergic neuron and GABAergic neuron. That red bar is for that data for that CUMS mice, and then green one is CUMS treated with lansomine. And you can see that green bar is very close to the actually blue bar. In fact, it is more than that one, but it's not significantly different from the blue one. So it is both for the glutamatergic neuron as well as GABAergic neuron. So just to summarize whatever I told you in this presentation, that in both CUMS as well as in chronic social defeat stress model of depression, we found there is a reduction in that neurometabolic activity as well as astroglial activity. And in the genetic model, activation of the forebrain excitatory neurons during postnatal life stress increase the anxiety, despair, and schizophrenia-like behavior. And that actually was resulted further into the enhanced activity of glutamatergic and GABAergic neurons in the cerebral cortex as well as in hippocampus. And with that Rasa-Sindoor intervention, before or after that stress, we can either preserve that behavioral and neurometabolic activity or ameliorate that neurometabolic activity to control level. And finally, that acute ketamine intervention or maybe subchronic lansomine administration restore that behavioral and neurometabolic activity in the social defeat as well as chronic unpredictable mice model of depression. I'm actually talking all these things because of that hard work of the group people which are here. And most of this, the data which I've shown here is carried out by the Praveen Mishra And that was actually started by Pandit Chelvan, now who is doing PhD in Netherlands. And later on, it was followed by Sampath Kumar. And I'm collaborating with Dr. Arvind Kumar, who is our colleague, and we are meeting every day, and Dr. Vidita Vaidya from TIFR. And we are supported from Department of Biotechnology, DST, and SCRB, and our parental organization, CSIR. And thank you very much for your attention.
Thank you, Dr. Patel, for finishing your talk in time. So we have time for at least one or two questions. So in chat, I think I could find, can you see the chart? I mean, somebody had asked which tissue sample is being used for NMR analysis, dissected brain tissue or cultured cells? When you say neuron or astrocyte, is it a cell suspension or the cytoplasmic fraction?
Yeah, okay. So I could not hear you in the first part because our microphone was out. Can you repeat again?
Actually, this is, visible in chart. If you open the chart, somebody has put a question for you.
Oh, okay.
So can you open your chart?
Yeah.
So you can read the question. This is some audience from fast distance.
Okay, so these experiments are done in the live animal. After, so we have to anesthetize the animal, or you can do the experiment in that awake condition. We infuse glucose for maybe 10 minutes or 15 minutes or maybe 90 minutes. And after that, we arrest the brain metabolism by freezing the brain in liquid nitrogen or using microwave in 1 second. And then we extract, take the tissue, prefrontal cortex. From there, extract that neurotransmitter or different amino acids which are present there by our extraction protocol. And then we analyze this extract by NMR spectroscopy. So measurement is done in vivo, but analysis is done under ex vivo condition. Next.
That's good. I mean, I think the audience who has asked, there is another. How does this RASA might work? Like you see the chart. I think there is another question for you.
Okay. Yeah. So this is actually, these are like Ayurvedic formulation, and Ayurveda actually believes on holistic improvement of the health.
Yeah.
So it actually overall improvement in the health. As I mentioned that there are some study done with Rasa Sindhu by Professor Lakhotia from your department. And using a Drosophila model, he has shown that there is a reduction in that
we have seen that paper,
species and all those things. And in fact, using fly model, he has shown that improvement of that health of those fly.
Okay, that is good.
Actually, I would like to ask you one question.
Yeah, sure.
So based on your NMR data, can you suggest a good biomarker for depression that could be used for patients?
That is a tricky question. For the biomarker, so far, like before, 10 years ago, when we were actually we're doing postdoc at Yale, and that time they were doing a lot of measurements for the GABA. And what consistently they found that GABA level in that depressed subject is lower. But that if you look at GABA level in epileptic patient also is lower. So it's not very specific for only for the depression. Now, further, when we started looking at the neurometabolism, we found that neurometabolic activity is very much low in that depressed subject compared to that normal person. Now, again, people will ask that, okay, neurometabolism is decreased in other conditions like Alzheimer disease or Parkinson disease. So, just only looking at neurometabolism will not be helpful, but neurometabolism together with other clinical symptom, we can use this one as a biomarker. But not only based on the NMR, we can say, okay, this person is having a depression or not. That will be, I think, difficult to say.
Thank you.
Dr.
Thank you, Dr. Patel, for wonderful talk. And
Okay. So I think every good thing also has to come some end. So Dr. Patel, thanks for a nice presentation.
I'm very sorry. There was some problem with my computer.
No problem. Actually, these are electronic hazards we face daily. So this has now become routine matter. So thank you, Dr. Patel. Because we have running short of time, so we will switch on to. So you may leave and so that we can switch on to another presentation.
You can stop sharing the slide, Dr. Patel.
I think I stopped already. I stopped.
No,
Yeah,
but
yeah, it's fine.
uh,
We can
stop
request the
share.
next participant
Yeah.
to start with the
Okay,
presentation.
so we are switching over to oral presentation back now, and second presenter is Dr. Binod, KIIT Odisha, India. Dr. Binod, are you online? Dr. Binod,
Here.
yeah, please. So go ahead.
Yes, sir, I'm here. Are you able
Sure.
to hear?
Yeah, sure. Share your slide and start straight away. So try to finish within 12 minutes. Actually, we are.
Yes, sir.
Ah.
Am I audible, sir?
Yeah, sure. Please go ahead. Okay, good.
So, greetings everyone from different parts of the world. First of all, I would like to thank the organizers for selecting my abstract, at the same time for giving me an opportunity to speak on this successful platform. So before going to the work, I would like to give a brief introduction about the circadian rhythm. I think you must have been already listening to different speakers for the past few sessions. So I will just have a quick brief. Like circadian rhythms, these are a component of the biological clock where you have a self-sustaining oscillator which can reset by itself, and this is going to have a rhythm for every 24 hours. So it is being maintained for every 24 hours. Okay, and hence it's been mentioned as circadian rhythms. Okay, so these are all having these different properties where they are endogenous, they are free running in nature, they are genetically regulated. So if you're going to have a brief idea like what these circadian rhythms are, if you're going to look at the activity of a man, you can actually see that you have the highest melatonin level being secreted during the midnight. Okay, then we wake up at the morning and then we have a good BP during the same time, and then we have a good concentration around 10 a.m. And then we tend to feel hungry at regular intervals. Okay, at the same time, we tend to go back to the sleep. So this is all in correlation with the environmental factors. Okay, so how is actually this particular thing being synchronized? Okay, so if you're going to look at the entire physiology, you have a master clock and you have peripheral clocks. So all of them are autonomous. They actually can perform their own activity. However, if you're going to look at the regulation with respect to the brain, you have a region in the brain known as suprachiasmatic nucleus, which is synchronizing with all of the other clocks through different innovations. Like you have autonomic innovation, you have body temperature, you have sleep, you have different social cues, and then you have secretion of this melatonin, which is one of the important factors. And then you have glucocorticoids also. So all of these, they are very crucial for regulating the synchronization process of peripheral clocks with the central clocks. Okay, and at genetic level, we have this BMAL and clock. These are transcription factors. They heterodimerize, they move back to the nucleus, and then they bind to several clock genes. So they bind to the EBOX elements of clock genes, and then you have secretion of PER, CRY, reverb alpha, and ROR alpha. So here we have positive and negative limbs. PER and CRY form a negative limb. So when they heterodimerize, when the protein products heterodimerize, they move back to the nucleus and hamper the BMAL and clock complex. Similarly, you have ROR and reverb alpha. The ROR elements, they actually are positive in nature. They give a positive feedback limb. So ROR is responsible for elevated expression of BMAL, and reverb is actually inhibiting the expression of BMAL. So this way you have positive and negative feedback loops been working together and regulating the clock process. Yeah. So coming to my hypothesis, we have this aging process where most of the biological phenomena get affected. So the melatonin, which is one of the crucial hormone required for the controlling of the clock process. If you're going to look at the melatonin levels, it's going to decrease by like around like 20, like 1/20th of the fold is being secreted at the elderly compared to a young kid. Okay, so with aging, we have oxidative stress, and with oxidative stress is associated with inflammation. This inflammation is being seen in several disorders, which are also linked with the circadian system disruption. So we have to see whether in peripheral system you have a role of control circadian system. So here we are actually looking at few inflammatory markers in the peripheral clocks. So the inflammatory markers we have chosen are COX-2, interleukin-6, one of the circadian clock reverb alpha gene, and SOX1. This is an anti-inflammatory marker. At the same time, we are also going to look at the effect of melatonin, exogenous melatonin during all the age groups. So for this, we have adapted this methodology where all the animals are being maintained at light dark 12-12 conditions. with lights on at 6:30 a.m. So this is the starting time of the queue. My queue here is the light, so it is 0. And then at 6:30 p.m. we are turning off the lights. So we are maintaining the light and dark cycle perfectly at 12:12. And then animals are divided into 3 different age groups. We have 3-month, 12-month, and 24-month groups to be studied. And in all the 3 groups, we are subdividing them into 3 further subgroups: control, vehicle administered, and melatonin. So we are giving melatonin at the rate of 30 microgram per kg body weight, administered along with 10% ethanol in PBS, physiological saline at ZT11. Okay, so food and water were provided ad libitum, and we had the dissected peripheral tissues at 4 different intervals, 2 intervals in a day and 2 intervals in a night. We have snap frozen the tissues, and then they are being stored for further uses. So all the experiments were carried according to the IAEC guidelines. So coming to the results, we are first going to look at the inflammatory markers. So here we can see in kidney, we have studied 4 different tissues: kidney, liver, intestine, and heart. So if you're going to look at the kidney, the COX, generally the COX2 levels are highest in kidney compared to the other tissues. So here we can see a slight rhythmicity, though it is not robust. We can see day-night variation. The levels are being highest in the day and the levels are being lowest in the night. Okay, but with aging, the so-called the phases are being altered. So the highest level, though they tend to continue in the daytime, you can see a shift being observed at the lowest level. And with respect to the 24 months, you again see the line is totally flat. And with the melatonin administration, we did not find much difference in the improvement. However, in 24-month group, we can see a restoration of level. In liver, we have seen a robust circadian rhythmicity where the highest COX levels are being observed during the night. The same thing is being continued in 12 months. However, in 24 months, we can see a phase shift, where we can see the reversal of the phase. The highest levels are being observed during the day and the lowest during the night. And with the melatonin administration, we have seen the rhythm is getting reversal in 3 months. In 12 months, the same thing is being maintained. And in 24 months, you can see a bit of restoration of the rhythm, which is respect to 3-month control group. So this is a very good improvement that actually we had found. And coming to the levels, the COX2 levels did not alter much with 3 and 12 months, but with 24 months, we found a significant change. We have found a significant increase in the levels, which are almost equivalent to the traffic control. And in case of intestine, we can see a robust circadian rhythmicity with highest levels being observed during the night. So here, actually, I'm mentioning a bar, I forgot to mention. So this is the light bar indicates light and the dark one indicates the night. And the black one indicates the control group and the red one indicates the melatonin treated group. So we can see a robust rhythmic pattern, and the rhythmic pattern is yet robust in 12-month group. But in case of a 24-month group, we can see a slight deviation in the rhythmic pattern. With melatonin treatment, though we did not find any much difference, and at the same time, we found the levels are getting dropped. In case of 12-month, we can see a robust rhythmicity. However, it is not that equivalent to the control group. In case of 24-month, we can see a robust rhythmicity, but the phases are not as good as the controls. And in case of interleukin-6, we can see a robust rhythmicity with the highest levels being observed during the night and the lowest levels being observed during the day. In 12 months, there are no rhythms at all. And in 24 months, we can see a similar pattern. However, you can see a change in the phases during the dusk and dawn. So the mean 24-hour levels, they are totally dropped with aging, and with melatonin treatment, we couldn't find any significant changes. In case of liver, the interleukin-6, actually, we had found a bit of rhythmicity in 12 months, but in case of control, 3-month and 24-month group, we did not find any rhythmic pattern. However, with melatonin treatment, we could see a robust rhythmic pattern, and it is been pretty significant in 24-month group. Coming to the intestine, in intestine, we have seen a slight deviation with respect to the levels. However, this is not that significant. Here also, though we are able to see that there is a lot of change, but because of the errors, we could not mark them as significant. So the same sort of behavior is being observed with respect to 12 month and 24 month. And with melatonin treatment, we did not find any change in the rhythm nor in the level. So we had seen that it has been arrhythmic throughout all the ages. Coming to the heart actually showed a very good rhythmicity with respect to interleukin-6. So we had seen that it is been really rhythmic with the highest levels being observed during the day. And in case of 12 months also, it's been highest, but however, you can see a phase shift. We have a phase advance been happening. In case of 24 months also, you can see the rhythmicity is as good as 3 months. And with melatonin treatment, we see disruption of rhythm and level. In case of 3 months, in case of 12 months, you can see a phase alteration being observed. And in case of 24 months, we didn't. It is as good as 3 months. We did not find any much restoration with respect to rhythm or level. Coming to the SOX, now that we had studied the inflammatory markers, we also want to look how actually these anti-inflammatory markers are behaving. So we had seen that the anti-inflammatory marker is in specific, like perfect anti-reversal pattern with respect to the COX levels in case of kidney. So here we can see the highest levels have been observed during the daytime, and then you can see a phase delay of 12 hours being observed. And in 24 months, the rhythm is totally lost. With respect to melatonin treatment, we had seen a slight change. Actually, the rhythms are getting, the robustness of the rhythm is getting lost with the melatonin treatment in young. But in 12-month group, you can see a bit of increase in the levels. However, there is no rhythmicity. The same thing is being found in 24-month group. In case of liver, we see a robust rhythmic pattern. This is totally reversible to that of what you actually have seen with respect to the interleukin-6 in case of liver. So we see that it has been highest during the night time. In case of 12 months, it has been phase advanced to 12 hours. In case of 24 months, we see a slight deviation in the rhythmicity, though it is not that rhythmic. However, you find a significant change with respect to the last time point, ZT18. And with melatonin treatment, you can see there is a phase reversal. You can see here, instead of getting peaked at ZT12, you are getting a peak at ZT6. So there's a phase advancement being observed. Though the rhythm is intact, you have seen advancement. The same thing is being followed with respect to 12 months. However, you can see a restoration of rhythm, which is equivalent to that of a control group. And in 24 months, also, you can see a bit of restoration of the rhythm compared to its counterpart. I mean, 24 months control groups, if you're going to compare with 24-month melatonin control group, the melatonin group is improving with respect to the rhythm. And in case of intestine, we haven't found robust rhythms. However, the highest peak has been observed during the night. And in case of 12 months and 24 months, there is no rhythmicity at all. But with the melatonin treatment in 3 and 12-month group, we did not find much changes. But in case of 24-month group, we have seen an increase in the levels. At the same time, we have seen that there is a rhythmic pattern. So this is almost a bit of restoration compared to that of 3-month control. In case of heart, we have seen robust rhythmicity of SOX1. This is actually following the pattern of the kidney. So we have seen that the highest levels are being observed during the daytime. And in 12-month group, we have a phase delay of 12 hours. And in 24-month group, again, it has been as usual as 3rd month. But in case of melatonin treatment, we actually have seen a phase advance where it has been advanced by 6 hours. In 12 months, the rhythmicity is lost. And in 24 months, it has been phase delayed. However, we can see the levels of SOX1 are pretty high in 24-month melatonin group, and they are actually showing rhythmicity. Now, coming to the circadian clock factor in kidney, we all the age groups actually showed rhythmicity. However, the rhythmicity is not robust. And with melatonin treatment, the 3-month group, we did not find any rhythmicity. In 12 and 24-month group also, whatever the rhythmicity was seen has been lost, and the levels were not restored. So the melatonin treatment is
wrap up your actually we're running out of time.
Sure, sir. Sure. I'll try to. And in case of liver, we have seen rhythmicity in all the groups, and it is being lost with melatonin treatment. In case of intestine, we did not find, however, it is not significant, but we can see that there is a change in the day and night variations of the river Vulva rhythms. And in case of heart, we can see it is slightly rhythmic, and it is more significant in case of 24-month group. And with melatonin treatment, we did not find much change. So coming to the summary and conclusion, we can see that with aging, these many inflammatory factors are being affected, with many of the factors being losing their rhythms at the same time altering their levels. And with the melatonin treatment, we could see many of the factors are restoring their levels, and few of them are actually restoring the rhythmicity as well. So thank you.
Thank you, Vinod. We can quickly move on to the next presentation in the interest of time. I think we... So next
So can I
presentation
stop sharing?
yeah. So next presentation is by Lakshmi B. Is Lakshmi around?
Hello, can you hear me, sir?
Yeah, we can hear you.
Start. Straightway start.
Okay. Okay. Yeah. Namaste to everyone. Thank you for providing me this opportunity to present my work on such a big platform. Today I am presenting part of my thesis work, neuronal protein profiling during post-embryonic development of Lepidopteran insect Bombyx mori and effect of phenoxycarb and 20-hydroxyecdysone on the same. We have selected Bombyx mori as a model organism for our study. In neurobiology, insects are being used as a model organisms due to various reasons. First one, here are few listed ones, like they have simple nervous system, less number of neurons, and comparatively larger sized neurons, easily accessible ones, with distinct brain function and behavior. Also, they have shorter life cycle that makes them more feasible for the various kinds of experiments to carry. Among various types of insects, we have selected Bombyx mori as a model organism because it undergoes a holometabolous type of metamorphosis. That means the juvenile form hatched out of the egg is a larva, which is characteristics of voracious feeding and crawling nature that transforms itself into a physically sedentary state called pupa that actually is metabolically very active, which ecloses out into sexually matured winged adult moths, which reproduce to continue the cycle. With this distinct post-embryonic developmental stages and different behavioral repertoires, Bombyx mori serves as a good model for proteome change studies. Apart from this, the aids in maintenance of culture and also Bombyx mori being a monophagous in nature, that means it feeds on a single type of leaf, that is mulberry leaf, and also semi-closed feeding system, that is only one stage of the post-embryonic development, that is larval stage, is feeding in nature. Hence, any changes that could interrupt the proteome interpretations will be excluded during this non-feeding stages of rest of the post-embryonic developmental stages. To attain this transformation in physical form, the central nervous system also undergoes a tremendous change with respect to its remodulation. As here we can see, the fifth late larval instar central nervous system is lengthier when compared to the rest of the pupal stage central nervous system and adult central nervous system. It clearly consists of brain, subesophageal ganglion, following by 3 thoracic ganglion and 7 abdominal ganglions, which forms 4 fusions, ganglion fusion 1, 2, and 3 between thoracic and abdominal ganglions, which further form a ganglionic fusion 4 to form compound ganglion 1 and compound ganglion 2, which will result in shortening of the length of the central nervous system in final stage of post-embryonic development, that is adult. So to attain this transformation, there are many important factors which will be helping each other to make a post-embryonic developmental stages to happen. Among them, major ones are the developmental hormones. Those are 20-hydroxyecdysone and juvenile hormone. The juvenile hormone is produced from corpus allatum, and it will be showing its effect based on the presence or absence of the 20-hydroxyecdysone, which is released from the prothoracic glands upon the action of prothoracicotropic hormone. As we can see here, the peaks of prothoracicotropic hormone will follow the peaks of 20-hydroxyecdysone. In presence of juvenile hormone and 20-hydroxyecdysone, the growth of the larva or any stage will take place, and the molting will be to the next instar, like larva will mold to larva. But when the juvenile hormone is not present and the 20-hydroxyecdysone peak comes up, then the larva commits itself for the pupal stage. Once again, in presence of JH and 20E, the larva will, pupa will continue its growth phase, and again, in absence of juvenile hormone and presence of 20-hydroxyecdysone, the pupa will commit itself for the adult phase. With this background, we wanted to know during this phase of transformation of central nervous system, there could be a huge amount of proteome change that would be occurring distinctly with each developmental stage. So we wanted to study that proteome changes with respect to these developmental hormones. The methodologies those were followed were different insect cultures were being procured from silkworm seed production centers Shadnagar, and they were being maintained in standard insect culture room conditions. The hormones administered were between 9 a.m. to 11 a.m. of the day, and phenoxyca, that is a juvenile hormone analog, was administered topically along the dorsal midline once a day.
Lakshmi, better you come to your result and conclusions. Come to your result and conclusions.
Okay, sir. Okay. So for O-Ferrous method was used for two-dimensional gel electrophoresis. These are the representative gels of our proteome analysis. For the upper panel represents the late larval instar. These are the second one is for late pupal instar, and the third one is for adult. The horizontal vertical panels are for control, phenoxycarb administered group, and 20-hydroxyecdysone administered groups. We have performed various analyses between the class matches and also between the mass set analysis, and we have come up with thousands of proteins. Those varied between the three biological groups, that is late larval instar, late pupa, and adult. Among those many number of proteins, we further performed factor projection analysis, and there were hundreds of significant proteins which were varied between the stages and also upon both the effects and 20-hydroxyecdysone administration within each stage of development. These significantly varied proteins were further studied based on their upregulation, downregulation, or newly expressed or disappeared categories between comparisons of within the biological groups and also upon hormone administrations. So among these various significantly expressed proteins, we have selected 18 robustly expressed proteins in all these categories and outsourced them for MALDI MSMS analysis. And here is the list of the proteins, those we got identified, and they were further grouped under 10 biological processes based on the gene ontology study. So we will discuss them here. The upper panel is for control, then second is for phenoxycarb, and third is for 20-hydroxyecdysone administration groups. Here, when we compared proteins between late larval instar to late pupa, we found mostly the structural proteins were either disappeared in their expression or they were decreased in their expression. As we can see, putative tubulin alpha chain and tubulin beta and alpha tubulin partial were almost disappeared, and phosphatensin was decreased in its expression, whereas autophagosome formation protein was increased in its expression with macro autophagy of dendritic cells protein RUF4 was increased in its expression. The immune response related protein complement C1q subcomponent protein was newly expressed in late pupa when compared to late larval instar. When coming to the comparison between late pupa to adult, we observed NFX1 type zinc finger containing protein which was newly expressed, which is related in neuronal development. And the metabolism related proteins like glutamate dehydrogenase and cellular retinoic acid binding proteins were either disappeared and decreased in their expressions. From late larval instar to adult, when we have compared, once again, the NFX1 protein was newly expressed in adult and the structural proteins were either disappeared. As we can see here, putative tubulin alpha chain was and phosphatensin was disappeared, and the alpha tubulin partial was decreased in its expression. Whereas the paramyosin, which is the motor activity related protein, was significantly increased in its expression, and autophagosome formation protein was also increased in its expression. Upon phenoxycarb administration, we have once again found that phenoxycarb has led to the disappearance of these structural proteins like putative tubulin alpha chain and phosphatensin.
Lakshmi, can you
Yes.
come to conclusion, please?
Yes, sir. Here only I was concluding. Anyways, I'll go to next slide. So, based on various observations, we are the first ones to generate the central nervous system proteome maps in the 3 decisive stages of Bombyx mori, that is late larval instar, late pupa, and adult of Bombyx mori. These can serve as reference maps for the central nervous system proteins in future studies. Study will also help in identifying markers of neuromodulation and neuronal development of Bombyx mori central nervous system. We have identified 18 robustly expressed, significantly expressed proteins, which we try to understand their role between various developmental stages and also upon hormone administrations. So the hypothetical proteins, those were identified from our study, can be further identified for their roles in the central nervous system remodulation of Bombyx mori. We observed differential effects of phenoxycarb and 20-hydroxy-ecdysone. We also hope our study for the new lead way to for the new hypotheses in the areas of pest management, better silk yield, or bioreactor generation, etc. Thank you very much for providing me this opportunity.
Thank you, Lakshmi.
There is
Thank
any-
you for finishing your talk. I think those who have any query, you may put your question on chat panel. So since we are running with time, so we will move to next speaker. So thank you, Dr.
Thank you,
Lakshmi.
sir.
Wonderful talk.
Thank you,
Very
sir.
interesting.
Thank you.
And questions for Dr.
Thank you, ma'am.
Vinod also could be posted in the chat box, and people can respond there.
Yeah. Yes. So next is Parul Gaur. Are you Parul Gaur? Are you online?
Yes, sir, I'm online.
Can we find Parul?
Yeah, she's there.
So
Hello?
Start
Yeah,
setting
Parul.
and straight.
Go ahead, Parul.
Yes, yes.
Oh,
Go ahead.
okay, sir.
And try
stick
to
to 10
restrict the time to 10 minutes now, because we are losing the time. You can keep the introduction limited and focus on your data.
Yes, sir.
Parul Gaur, are you online? Is there any problem?
She has kept her mic is muted.
Actually,
Yeah, she's sharing.
internet connection is not over here, and I'm not able to share my slides using my laptop, and so I'm trying to share slides with my phone.
Okay, okay.
That's the problem, sir.
Dr. Parul, I think, can we move on to next speaker? In the meantime, you arrange your talk. Sure, sure. So, thank you. So just wait after 10 minutes. So we are moving to next talk.
Okay, sir. Okay, sir. Sure.
So, Dr. Naveen Sendilathan from Colombia. Are you? Yeah, good.
Yeah.
So share your slide and try to stick to 10 minutes, please.
Okay, thank you. Okay. Can you see the screen?
Yeah.
Okay. I'm going to start. Just 1 second. Okay. So thank you so much for the organizers for organizing this and allowing me to share my results with everybody. So I'm going to talk to you about what happens in the brain when two movements are planned simultaneously or together. For example, when you play the piano or when you make sequential eye movements, multiple movement plans are computed by the brain together, but then they are serially executed. So how does this computation happen in the brain? So I'm going to use eye movement as an example for this whole for this whole study. When we see a visual scene, the information goes to the back of the brain, and then the brain chooses some stimuli to look at, to attend to, and make plans to make eye movements to those visual features. So one type of eye movement is called saccades, which are the rapid eye movements that we make every day. And I'm going to be talking about saccades in this talk. So again, how are saccades computed in the brain? A very simplistic view of how saccades are computed in the brain is once we see, first of all, there are so many areas in the brain as shown here, which are involved primarily in visual selection and vision and eye movement planning. So specifically, when we see some stimulus, the information goes to the visual cortex in the back of the brain, and then some areas in the brain processes information and then provides input to the what pathway and the where pathway. And more areas further processes information and then give the information to even upstream areas like frontal eye fields. And the frontal eye fields produce a movement plan out of this visual feature extraction and provides the information to superior colliculus. And the superior colliculus sends a feedback back to FEF, and then it finally provides the information to the eye muscles to actually make the eye move. So this is a simplistic view of how the eye moves after we see something. We know for from years and years of literature that frontal eye fields, FEF, is involved in generating saccades, these eye movements. But the topic of this talk is what happens when two movements or multiple movements are planned together. So to study this, we used monkey models. So we trained monkeys to fixate on a central fixation spot. And then we showed a target, and then we showed another target. And the time between these two targets is a TSD, which is target step delay. This could be short. They could appear very quickly, or it could be medium, or it could be long. 3 conditions. And after we showed 2 targets, the monkeys have to make saccades sequentially. So the first to the green and the next to the red. So the first and second saccades. They look like this. The first saccade would look like this for each of the 8 positions from center out. And the second saccade will start from that out, and then it will go to other position like this. Okay, so what does the neuron look like? What does the neural activity look like? So to study what happens in the brain, we can have 2 approaches. One approach is we just record from the neurons in FEF, from single neurons, one neuron at a time. And then we can see what happens, how this neuron fires on each trial. So on the first trial, it fires like this. On second trial, it fires like this. And then you can keep going. And we can look at the trial averaged activity. And that's one way of looking at this mechanism. The other way is recording, if we do this, then the neural activity looks like this. So for example, for the short TSD, I'm aligning the activity of the neuron with T1, the first target, and the second target. So for the first target, it increases, and then there's a small plateau, and then it again increases. And then you can see the same thing for the second target also. For medium TSD, it just increases. There is no plateau, there's no stopping, and then there is increasing again. It just increases directly. And for T2 also, it increases directly. And for T2, it's earlier than T1. For medium, it's earlier than short for T2. And for long, we see the same pattern again. It's earlier, and then it just increases directly here. So this is what we see if we record from single neurons at a time, and then we just average across all the neurons. The other way to do it is to record from multiple neurons at the same time, and then just do fewer number of trials. For example, in one trial, if we record from all the neurons, then we have neural activity from all the neurons, and we can average across all neurons. This will give us a trial by trial information of what's happening in the brain, and that's easier to look at that is necessary and easier way of looking at the mechanism of what happens on a trial by trial basis. So just to summarize, we have two methods, recording from one neuron at a time and averaging across trials, or recording from multiple neurons at a time and averaging across one trial. So we have this information, but we would like to have this information to look at the mechanism. So how do we go from here to here? So there is a mathematical way around it, which is called dimensionality reduction. I'm going to very briefly talk about this method before going into the results. So imagine there are like multiple neurons, neuron 1 to neuron n, and I'm plotting the neural activity here. So gray and green are two different conditions. You can think about it as T1 and T2, two different conditions. So I can do a transformation called PCA, which is principal component analysis. This would extract those signals in the brain that are dominant across all the neurons. You can think about this as a high dimensional averaging. So it will extract those signals and it will create these principal components, PC1, PC2, PC3. So now I can see how many components do I need to replicate this entire system. So I can plot the cumulative variance explained, and it looks like I just need about the first three principal components to explain about 90% of the variance. So I'm just going to plot these three PCs with respect to each other in a 3D plot, and it'll look like this. So I'm reducing a dimension of n to just dimension of 3. So I'm reducing the dimension from a high dimension to a low dimension space. Now I can look at how the neural activity follows in this 3-dimensional space. This is called a neural trajectory. So now I can compare these two conditions in the low dimension and see how similar or how different they are and look at the mechanism. So if we do this analysis for the data, then for short TST condition, T1 and T2 looks like this. And now I can see how similar they are. So for this, I calculated a parameter called subspace sharing. So how much the subspace is overlapping between these two. And it looks like there is very less overlap. It's about less than 50%. Now, when we look at the medium condition, the overlap is higher. And for large TST, the overlap is maximum. So this makes sense because if two plants are planned very close to each other, then they are forced to be in different subspaces so that they can parallelly evolve and then they can be executed. But if they are already spaced out in the long condition, then there is no need for them to be in two different subspaces. They can reuse the same subspace. So subspace sharing is higher. So now we want to look at the causality. Like, so what is actually happening, and why are the neural activity forced to be in two different subspaces? For that, we did a simple computational model. So we did a simple neuronal model where we showed, after we showed the target, we have a tuning curve which represents the spatial information. So how much neural activity is present in each of the 8 positions. This is one such example tuning curve, which says that this neural activity is maximum in around 2nd or 3rd position, and then it falls down in a cosine way. So this gives the spatial information of the neural activity. And then we have the temporal information, that is what happens through time. So the neural activity increases after a target and then it decreases later. So then it will look like this for an example neuron. So if we multiply these two, spatial and temporal, and then add a little bit of noise, then we get the actual neural activity, which is shown here. So now we can use this model for T1, and then we can use this model for T2 also. So we have 2 different models, how the neuron will fire after T1 and how will the neuron fire after T2. Now, what we can do is introduce a small inhibition term. So we can make these two mutually inhibit each other. on a trial by trial basis. And the power of this inhibition varies inversely with TST. That is, if the two plants are closely planned, then there'll be higher inhibition. And if it is spaced out, then there's lower inhibition. So after this inhibition, without going into details, the neural activity will look like this for T1, that is lower in activity, but delayed in time for T2. So the inhibition is in two different ways for T1 and T2. So how does the neural activity, how does the model do compared to the data? So this is the data that I showed you before for T1 and for T2, and the model looks like this. So it replicates the data pretty well. Now we can look at the low dimensional space and see how that works. So in the low dimensional space for short, medium, and long, the neural trajectories look like this. Now, when we calculate the subspace sharing, it looks pretty much similar to the data. So it's low for short and a little bit higher for medium and the highest for long. So just to summarize, the neural data and the model with inhibition have similar properties. Now, we try to remove this inhibition and see how does the neural activity work without the inhibition. So if we remove the inhibition, all these things go away, and then the neural activities would look like this. So again, this is the data, and this is a model without inhibition. So they look completely different. And again, as expected, if you look at the low dimensional space, they will also look very different. The subspace sharing is very high because there is no inhibition. So without inhibition, the model looks like this, which is very different from data, which means that what is actually, in fact, happening is there is mutual inhibition between the two plants, which are happening very close to each other. And if they happen farther from each other, then there is less inhibition between these two plants. So just to summarize, FEF, frontal eye field, is involved in planning sequential saccades or sequential movements. And these two sequentially planned saccades, the plans mutually inhibit each other if they're very closely planned. So this is like a capacity sharing model. There's limited capacity in the brain, and two plans are fighting for that capacity. And the plan that was planned first get executed first, and then the next later. But they can both evolve together in parallel, but they can be executed in serial. So with that, I would like to thank Deblena, who collected all the data, and this is part of her thesis work. And Adi, who was my supervisor, who helped me navigate through this project, and DST for the grants, and IASC where the work was done. Thank you.
Okay. Thank you, Dr. Naveen, for finishing line. So if there are any questions, you may use, or anybody can use the panel and ask questions to Dr. Naveen. In the meantime, let us move straight to the speak. Presentation to Dr. Naveen.
Sorry, I'm not able to hear you properly. I'm sorry. Can you repeat? Sorry.
Hello.
Hello.
Yeah, Dr. Parul.
Yes, sir. Sir, so can you see my screen?
Yeah. You can go to full screen.
Okay. Okay, sir. So first of all, my namaskar to all in one present at various time zones on this earth. And I thank you for providing me a chance to share my small piece of PhD thesis work with you people. The topic of my today's presentation is variation in measurement of neuronal soma size associated with reproductive differences in our seasonally breeding male via weaver bird, Plosis philippinus. Moving forward, firstly, I would like to discuss about my experimental animal. They are small, sparrow-sized, social and gregarious passeriforms breeding colonially, inhabiting hot, dry countries and abundantly seen in Indian subcontinent. Breeding male typically bears a yellow marking at crown, is polygynous and makes architecturally amazing retort-shaped nest during breeding phases, which in their lifespan, which we can see in our vicinities also. Their nests are elaborately woven and ingeniously constructed. The choice of nest location is such which may defend them from their enemies. Moreover, the male bird also attracts the female by hanging upside down from the nest while calling, which changes during its breeding phase, and fluttering his wings. A female selects the male only after observing the quality of the nest. Thus, we can say that a proper nest is an active resource necessary for successful breeding. Moving forward to the research material, that is brain and specifically the neurons, it has been reported by many researchers that neurons are not only essential for learning, but also in memory formation, behavior, cognition, navigation, social sexual behavior, and many more. Neurons are plastic structures and show differences according to the different environmental selection pressures, and also appear to be controlled by some heritable mechanisms involved in neuronal development. Thus, differential demands create differential selection pressure and over animal, and for coping with it, there arise differential neuronal parameters. And when we consider these neuronal parameters, neuronal soma size also appears plastic in response to environmental conditions. Moreover, it is assumed to observe a larger dendritic tree, thickness, more synaptic connections, that is spine density and morphology, and higher neuronal activity. Bridging the gaps between basic and clinical neurosciences, the results of various clinical researches have shown that due to prevailing high degree of plasticity, these structures of synapses and the resulting functions change throughout life. Cahal discovered dendritic spines, and today we all know these spines to be the major sites of excitatory synaptic transmission. Clinical neurosciences have shown that various diseases are also associated with neuronal plasticity. In case of birds, it has been shown earlier also that avian brain exhibits seasonal plasticity in response to photoperiod and hormones. So, keeping it in mind, the differences in social sexual behavior of male bare-weaver bird, we designed the research study as finding seasonal variations between different parameters. To do so, we used two staining techniques. First was the nasal staining crystal wallet technique, which was utilized to find the cytoarchitecture of brain, and the second one was silver impregnation Golgi Colonia method to visualize the neurons. After the routine processing of brain sections, the photomicrographs were taken using computer-aided Nikon Eclipse TI camera, and further, the measurements were done using Adobe Photoshop software. The spine density was calculated using formula given by Feldmans and Peter. GraphPad Prism software was utilized to compare the statistical values, and 12 animals were taken for this study, and all the above mentioned protocols were carried out in accordance with the institutional animal care guidelines. This photomicrograph shows crystal stained section of brain of bare-weaver bird, revealing the layered structure showing more superficially lying hypopilium apicale, followed by interstitial nucleus of hypopilium apicale, hypopilium intercalatum, and hypopilium densocellulare, which can be seen ventrally delimited by the mesopilium. This plate shows photomicrographs and camera lucid drawings of neurons of three types of neurons observed in Golgi Colonia method. The most abundant were the multipolar neurons. A and A prime shows the non-breeding state neurons, and B and B prime shows the breeding state multipolar neurons. C and C prime shows the non-breeding state pyramidal neurons, and D prime shows the breeding state pyramidal neurons. C and C prime shows the non-breeding state stellate neurons, while F and F prime shows the breeding state stellate neurons. When the statistical observations were analyzed, we found significant seasonal fluctuations regarding various parameters of neurons in HA region, which included increased neuronal soma size, increased dendritic thickness, increased spine density, and plasticity in dendritic spine morphology. I'm going to discuss each pair separately in the following slides. This bar graph shows an increase in soma diameter during breeding season, which is indicated in red bars. The larger soma size may be assumed to observe enhanced spatial memory and other related aspects of neurons. Size of soma may act as an indicator of neuronal activation, function, and differential response to environment. Increased soma size can be attributed to production of more substrate, that is the sex steroids, by engaging in more metabolic activities. Thus, during breeding season, more concentration of hormones can be related with increased soma size. Moreover, highly metabolically active neurons show dependence on more mitochondria to satisfy their energy needs, as these mitochondria originate in the soma of the neurons. The same thing was also shown by Dr. Patel in his presentation, that neurons contribute to 5% of the body weight, but 20% of the energy consumption is done by the neurons. And so when an animal is in an aroused state, then more number of mitochondria is also required. So it can be owed to the increased soma diameter also. All these selective adaptations or variations in soma size can be attributed to the other related aspects which follow. This graph shows seasonal variation in dendritic thickness, with again the red bars showing significant increase during breeding state of animal. This increased thickness of dendrites may be related with an adaptive mechanism of the neurons, especially dendrites, to support an increased number of spines over them. Many researchers have shown these dendritic shafts to act as post-synaptic elements forming synapses with the other axon terminals. This graph shows seasonal variation in the density of spines being significantly more during breeding phase, again shown in the red bar. This increased spine density favors the view that HA neurons receive strong retinal inputs and stream them to the following telencephaltic structures. It is well established now that spines act as a site of memory formation, thus their plasticity will also lead to seasonal differences much required during breeding season for socio-sexual alertness. Thus, the fluctuations in photoperiod and variations in gonadal steroid levels might be held responsible for a more complex neuronal network during breeding phases. They affect the soma size, which further influence dendritic spines and reproduction. During breeding season, when a male is under performance pressure for mate selection, its socio-sexual activity changes, which may be related to an increase in the number of inputs received by individual neurons, either by increasing the density of the spines or by increasing the length, number, or thickness of the dendrites. For all this purpose, the neuron soma size should also increase. Thus, these increases indicate the idea of more visual burst use during breeding period to surmount the provoked conditions attributed to the presence of a female partner and increased hormonal levels. Thus, HA neurons can be assumed to be inducing and regulating the various aspects required to transform a non-breeding animal into a breeding one. Thus, to summarize, I can say that seasonal fluctuations in gonadal steroid levels directly or indirectly mediate the neuronal morphological plasticity. Differential selection pressure and demand leads to increased soma size during breeding season. The bigger soma contains larger cellular and metabolic machinery needed to serve a larger and thicker dendritic tree and more synaptic connections, that is increased spine density and activity. Thus, it can be assumed that neuronal soma size could be reflective of memory and learning ability of an organism, with larger neurons supporting with larger neuronal soma supporting various related aspects of enhanced memory and learning ability. Thus, to conclude at the end, I would like to say that the survival advantage and disadvantage well regulates socio-sexual behavior of an organism. For example, like in Baya, which breeds during monsoon seasons, it has an advantage of good water and food resources in India, as in India the monsoons are the time for kharif crops, good food supply to the birds. Thus, positive selection results in increased soma size, dendritic thickness, and spine density, favoring the view that postsynaptic receptors increase in visual bursts to receive a heavier input of incoming information. These increased parameters can be assumed to make an animal ready to withstand changes occurring within and around it during the breeding state. At last, I would like to acknowledge the funding source, the UGC, for providing me support to carry out this work. I would also place my high gratitude towards my mentor and guide, Professor Ifi Srivastava, who was always there to support me. I would also thank my lab mates over there during my research tenure. And once again, I would also like to thank the organizers for providing me this platform to share my work. Thank you for patiently listening to me at this night time. And any of the questions, if there are welcome.
Okay, thank you. Thank you, Dr.
Thank you,
Parul.
sir.
Very wonderful presented your work. That's very good. So since you are the last speaker of the session and we are really running out of the time and therefore we cannot ask questions at it, but you can see the chart panel, you have seen so many good comments. So you may go through with that and then you may respond on that. So with this, if Dr. Sandhir is online.
Yeah, I'm online. Yeah,
Yeah,
online.
sure. So
And
I
I would say
request
that
Dr. Sandhir, my
yeah.
co-chair, to conclude the session, please.
So I would say we had a wonderful presentations starting from Dr. Patel, who talked about NMR, how it could be used in chronic stress disorder, and we had young presentations, all excellent from different covering different aspects. I think I enjoyed the sessions, and there is enough to learn from young presentations as well as from Dr. Patel. And I will thank Dr. Trigun for conducting the proceedings so well. And I was virtually on a holiday, I would say, listening to only the presentation part. Thank you, Dr. Trigun, and all the participants for making this a great session, I would say, and that too at odd hour of the evening. And take care. I think we have the closing ceremony starting in some few minutes. So bear with us for that we did not have elaborate discussion because of paucity of time. Thank you once again, everybody.
Thank you, Dr. Sandhir. Now it is over to organizers, Dr. Anita. You may take over and your team may take over for the session.
Hello.
थोड़ा ठंडा ज़्यादा हो गया मैंने 23 करा था बीच में गर्म हो गया था कोई नहीं बिना देखे So a very good afternoon, a very good evening, and good night, and early morning, good early morning for different delegates from different regions. Madam Gauri Devi, Madam, Namaste. And I hope Dr. K.K. Deepak sir has also joined. Okay, so
I think not yet, not yet. Yeah.
So can we call?
I'll call.
I'll ask some volunteer.
मैं कर देती हूँ आप शुरू करो
Start करा दो? Okay.
शुरू करिए मैं कर रही हूँ उनको फोन
अच्छा आप कर रहे हैं thank you. No, let me see. So, like every good thing has to come to an end, and we do not know how the 4 days from 4th to 7th October have gone. And first of all, I would like to give the welcome address. Once again, I begin with thanking.
Okay, just take a minute. I was just waiting on.
वो वाला था. अच्छा इधर देखा. Video.
I hope so. Video is on. You can start.
Okay. So, the special respected dignitaries, Professor Apparao Podili, Vice Chancellor, University of Hyderabad, Professor P.N. Tandar, Founder, President, NBRC, Professor T.R. Raju, Dean IAN, and previous Dean Professor M.K. Thakur, past Dean, and Professor Ishan Patro, present President, and all the present and past EC members, Professor Vinay K. Khanna, Secretary Headquarters, IAN, Professor Pankaj Seth, General Secretary, IAN, and Professor S. Dayananda, Dean SLS, University of Hyderabad, Professor P. Prakash Babu, University of Hyderabad, Professor P. K. Seth, one of the founder of IAN, Nasi, Senior Scientist, and Chairman ILSI India, Professor Padma Bhushan V.S. Ramachandran, Professor Suresh Ratan, University of Aarhus, and Professor William J. Schwartz, Texas, and Dell Medical Center, and all our eminent neuroscientists, colleagues, friends, students, and volunteers. It has been our distinct pleasure for myself, Anita Jagota, and Dr. Shashi Bala Singh, co-host and president of IAN till date, to welcome you all to the validatory function of the IAN e-conference and 38th annual conference of the Indian Academy of Neurosciences with theme Basic and Clinical Neurosciences, Bridging the Gaps, is being jointly, which was jointly organized by Department of Animal Biology, School of Life Sciences, and Institute of Eminence and the National Institute of Pharmaceutical Education and Research, NIPER, Hyderabad. Once again, I would like to thank Professor Appa Rao Podili, our Vice Chancellor of University of Hyderabad, who has extended full support to me, not only during the preparation and once the idea of organizing the physical conference 2 years back had originated, through the journey when it got converted to the e-conference, and the last 4 days, he has been throughout a big support to me. I would also like to thank Professor S. Dayananda, Dean School of Life Sciences, University of Hyderabad, for his full support during the preparation and during the conduct of this conference. I would like to now welcome an guest of honor who has already joined, Dr. M. Gauri Devi, Madam, and Chief Guest, Dr. K.K. Deepak, for their valuable time and accepting the invitation to attend this ceremony. Every year, a large number of neuroscientists from across the world come together and contribute to the success of our annual meetings. This year, however, owing to the unprecedented circumstances of COVID-19 pandemic, we were unable to hold the conference in person, though we were getting ready as traditionally done. However, with the gift of technology, support, cooperation, and coordination among the entire organizing team, IAN, speakers, and participants, I think we can say that the conference has been a resounding success. The conference participants comprised of distinguished scientists, experts, PhD scholars, postdoctoral fellows, postgraduate and undergraduate students from over 20 countries. It was really heartening to see the symposiums in which we had speakers from UK, USA, Japan, Switzerland, and India in a single symposium, delivering lectures back to back from comfort of their homes or offices in different time zones. Because of the virtual nature of the conference, we were able to have many international speakers to add to the neuroscience feast we had on offer. Over 300 delegates heard lectures by 73 symposium speakers, which included, in addition to 10 special lectures, we also had 44 poster presentations and 25 oral presentations. Keeping in mind the diverse backgrounds of attending delegates, the sessions had been planned to satisfy the scientific temperament of all the participants involved. We had opened the special lectures for the entire neuroscience fraternity through live streaming on Facebook and YouTube, and the symposium on mind body medicine had a viewership of over 1,500. From our end, we tried to ensure that the conference was conducted as smooth as possible, but some people might have faced inconveniences due to connectivity issues at their end, which created glitches at times. We regret for that, and now I take this opportunity to invite Dr. Pankaj Seth to give Secretary's report.
Good morning, good afternoon, good evening to the participants who may be attending the valedictory. We have delegates from US, speakers and chairpersons from US and different countries, including from US to Australia in all different time zones. First of all, it's really a great honor that Ma'am Professor Gauri Devi has joined us for this valedictory function, as well as Dr. Professor KK Deepak, who have been, supporting this organization in different ways. As far as this organization is concerned, this academy is growing both in terms of its numbers of delegates as well as number in its academic activities. This has been possible mainly because this academy grows like a family. We have people of different diversity, and this academy has grown not only at national level as well as at international level by its recognition by organizations such as IBRO, FAONS, Japanese Neuroscience Society, as well as SFN, which also has a Bangalore chapter with the help of people at NIMHANS Bangalore. So with that, I would like to just in brief remark, this has been repeated several times during the meeting, that our current membership holds of life members is around 1,447, and we have added 69 new life members in last year, and we have several others, around 70 associate members, and the academy is growing. The activity at different chapters, there are 7 local national chapters in India, including 2 in abroad outside India. One is at Chicago, another one is in Japan, and US and Japan, and which simply shows that our members and our active members are basically spread out all over the world and are knitting this society and this academy together. We are very fortunate to have some of the founder members with us. We, like Professor Seth, who's been guiding us on different occasions. We take his guidance on various matters, which are always very useful. We had, at the inauguration, we had Professor Tandon's, blessings to start with, and now we have Dr. Gauri Devi's, a very, accomplished neurologist of international standard. And ma'am has been at the director of NIMHANS and has held so many portfolios in various capacities. Her work on Japanese encephalitis has been, known very well throughout the world. So we have, this is what we are as society. And I'm glad to announce that, our members are doing well. We are having a lot of good publications, program grants, different awards. So this academy is growing, and which is a very healthy sign. With that, I will, give the floor back to Dr. Anita, and I'm willing to accept any suggestions and, you're on the website, on the way we are outreaching, and just let me know on my email, and we'll try to introduce those things as well. Thank you.
Thank you, Professor Pankaj, and this gives me immense pleasure to invite Professor M. Gauri Devi from EBAS, New Delhi, India, and this audience knows everything about the Gauri Devi Madam, how established, outstanding neurologist, neuroscientist, and researcher Madam is. And because we were running behind the time, I would hand over the platform to the speaker, who is the guest of honor, Professor Gauri Devi Madam.
Thank you very much for this very kind invitation to take part in the closing ceremony. Professor Shashi Singh, President of the Indian Academy, Dr. Pankaj Seth, the Secretary, Dr. Deepak, Chief Guest of the function, and Anita Jagota, who gave a very nice review of the whole thing. The organizing secretary must have worked very hard for 2 years, and Dr. Khanna and Dr. Khatri. You know, the very emblem of the association says, bridging the gap between basic and clinical neurosciences. I think this is what we have been doing all these years and propagating this idea. Now, at the moment, we are all seized with the pandemic of COVID-19. And as a neurologist, including our colleagues across the country, we are really astonished and surprised that so many neurological disorders have been reported in the short period of about 6 months in COVID-19. We have to know the various mechanisms. Some work is already being done. But if you look at the proteon manifestations, the spectrum, it seems to involve almost the entire neuraxis. From the brain, we have encephalitis, encephalopathies, acute necrotizing encephalitis, seizures like epileptic attacks coming on, and then we have other disorders of the peripheral nerves, the Guillain-Barré syndrome, cranial polyneuritis, then coming to muscle disease, myasthenia, association of other disorders. Every day the list seems to increase. Apart from ageusia and anosmia, which are well known. Now, the neurological manifestations, particularly the strokes due to the coagulation defects and the problem with the vasculature, are increasingly recognized, not only in the elderly, but also in the young people. So these are challenges, and these disorders, although the treatment may remain like the usual neurological disorders, but being complicated with COVID-19, our approach should be different. And it's really surprising that these neurological disorders associated with COVID either antedate the COVID-19, that means every neurologist should be alert if they see a neurological disorder. Otherwise, we would have given a diagnosis. Now we start looking for COVID-19. It can come, these disorders can come along with the acute onset or even follow after a week or 10 days. So you see the whole picture seems to be enlarging day by day. Now, when we are discussing this in this forum, obviously it's a great opportunity and a challenge for all the neuroscientists, the very mature, the growing young, and the students, to look at the various mechanisms. Why these are occurring, what are the various mechanisms? And if we knew that, we would be able to target the drugs, the receptors, and so on, and offer new and novel methods. As a clinical neurologist, faced with this COVID-19 neurological disorders, I'm looking up to this organization with brilliant neuroscientists across all age groups. Take it as a challenge and give us the answers, the mechanism, how does the virus go, what happens to the immune system in the brain, how do we tackle it? There are so many questions, and I hope I will be able to see this in the next couple of years, because the way the publications are going on, the research is going on at a supersonic speed, literally. I think we look up to all of you, the neuroscientists across the country and, of course, internationally, to give us the answers and help a clinical neurologist at the bedside to help the patient. Thank you very much. It was a great conference. I've been listening to talks in between. I thank the organizers for giving me this opportunity to give the closing address. Thank you.
Yeah. Yeah. Thank you, Professor Gauri Devi, Madam, for your very enlightening address. And I now request Professor K.K. Deepak to give his address as chief guest.
Good evening, everybody. So, am I audible?
Yes, yes.
Yeah, okay, thank you. Thank you, Dr. Jagota. I appreciate the way you have organized this conference for a couple of days. I've been seeing the program and it's a wonderfully held conference. And this was held during the COVID time, and that's too commendable. So I cannot see the dais, but I can imagine who people are on dais. I respected Dr. Gauri Devi. So I always respect her and in terms of the contribution to the neurosciences. Dr. Shashibala Singh, Dr. Vinay Khanna, Dr. Pankaj Sheth, Dr. Dharmendra Kumari Khatri. So with due respect to all people on dais and off the dais and those who are listening at this time of hour, which is very unusual for the conference, but thanks to the COVID, we are doing innovation. So I'm very happy during the COVID, you could organize such a wonderful conference, an exemplary conference, and across the countries, like people are there in the USA who are listening and maybe in Japan, those who are listening. So again, I thank you very much for the organizers and those who have done a wonderful job from the point of view that's doing science. Now, considering IAN has been doing wonderful work, so we were involved last year and we listened that it's a great work going on. Now, let me come to the 2 points which I would like to share with you. During the period of COVID, 2 important things have happened in the country. And 2 important things have happened to the country in order to make our science strong. The neuroscience is also a science to make neuroscience strong. So I'll come and discuss how about doing this. The issue with the country has been that we are only advancing in the field of space and atomic research. And this happened so because there was a denial for doing bird science or bird technology. The moment 83, India was denied not to have any nuclear program, and then India has bounced back. So happened with the liquid engine fuel with the space research, and we see the both science are aggressively going ahead. Now, is it happening in neurosciences or health sciences, or for that matter, other sciences? Take agriculture. So the reason has been, if you borrow a technology, the technology is old. And if you do research on technology, your data is going to be a repeat performance. Because by the time you get a technology, it could be patch clamp, or it could be muscle nerve activity recording. The technology is already 10 or 20 years old. Therefore, a common sense research might have already been done. Now, what to do, how India can handle this problem? So for that reason, during this COVID period, exactly the 4-5 months, India has bounced back with the policy called STIP, S-T-I-P. This is the 5th science policy which is coming up. It's called STIP 2020. I'll not go into further detail, but I'll just touch upon what it's doing. The final draft would be out in a couple of weeks, this week, and this final draft will be available to you all to give your input. This envisages how can we do innovation in science and technology. The I is underlined, and it believes that all youngsters have imagination. They should come up with the imagination and form and give a new idea which can take up a science of a new technology, Indian-owned technology, a new kind of imaging tool or new kind of analysis tool or new kind of a device by which you can generate your own data. Because if technology is existing, it is absolute obsolete technology. So you have to innovate your technology, have your own equipment, the way Dr. Painter devised his own equipment and then did research continuously on his own equipment. He did not bow about technology. So that's what I would envisage, that youngsters should come with the innovation in science and technology, including neurosciences, and generate a new idea, convert into a product, patent, or procedure. And that would be the humble request to all youngsters and all people to encourage that at least come with one patent in the field of neurosciences, and then you advance. And that's how India will go up. So this is one thing which has happened during the last 4 months. STIP 2020 is on the way. It is there, and in one month's time, it will be put on the table in the house. Therefore, let's all come together and use how this can be used. And there are several features of this technology, inclusiveness and other things, but important is doing innovation. So let's all pledge that at least the youngsters will come up with one patent, at least one patent, and then go ahead. Those who have quality, they can do it along. The other thing which has happened, by which you can make yourself strong, one is do strong science, but however, for that you have to have a strong brain and a strong body. The government of India has come up this last couple of months, only the last 1 year, they have come out with the Fit India protocols. And these are fairly detailed protocol for all age groups to make your both body and mind happy. The way you had a mind-body program here, how to make body and mind happy. India has come out with Fit India protocols. These are available on the Fit India website of Government of India under Sport Ministry. And I wish that each one of you make yourself fit in terms of the physical fitness, mental fitness, and therefore you can perform innovations. So I've got two messages for you. One is follow STIP 2020 technology policy. Give your inputs. Please write letters or make a forum or do a discussion, or maybe IAN should have an in-house discussion what kind of a science, technology, innovation policy is required for doing neurosciences in the country, period. That you can do as a focus group. You can give a group suggestion from IAN to the ministry. There are two houses there. The Prime Minister, Scientific PSA, Principal Scientific Advisor to Prime Minister, and also there is Secretariat in DST. So that's what we can do. Second thing, make yourself fitter from the mind-body point of view and come up with a new. And for that, you can always visit. So with that, I come to an end, considering this time limit and other things. I thank you very, very much for the organizers for doing wonderful work, and especially in so difficult times. And with that, I close my talk, and thank you very much once again inviting me for this closing ceremony. Thank you so much.
Thank you, Professor Deepak, for your address as chief guest. And now I request Professor Vinay K. Khanna to give his address. And I'm sure the students must be waiting for your address.
Thank you, Dr. Anita, and very good evening to all of you, and those who are in other parts of the globe, to them, good morning, good afternoon. Respected Dr. Gauri Devi, Dr. K.K. Deepak, the President of IAN, Dr. Shashivala Singh, my colleague, Dr. Pankaj Seth, and the dynamic organizer of this international webinar, Dr. Anita Jaguta, Dr. Dharmen Khatri, all those who are sharing this virtual dais. See that, as Dr. Anita had mentioned in the beginning, that every good thing that comes to an end. Before I announce the result, I'll just take a minute just to appreciate the endeavors of Dr. Anita Jaguta and the President of IAN, Dr. Shashivala Singh, to have this successful experiment to have the international webinar. I mentioned it earlier also. This is in the history of Indian Academy of Neurosciences. This is the first time that we are having. And this was the successful experiment. I congratulate you all and the team who has been really involved, and they have worked day and night for the success of this. Besides having the interesting presentations from the distinguished neuroscientists from India and different parts of the world, this webinar attracted many young researchers from India, and moreover the fact that young researchers from MIT USA and University of Bonn Germany, they also took part in the award session. We all enjoyed the interesting presentations by the participants and even have interacted with all those who participated in the poster session, which was quite interesting and was thrilling for all of us. The quality of the presentation was excellent, and it was quite tough for the referees to take a decision also. But as the number of awards are fixed, it was definitely a challenging task for the members of the committee to take a decision. I have just received the compiled scores a few minutes back from the committee and have great pleasure to announce the result for which my young colleagues will be interested in more. As you know that we have awards in the poster session. There are awards in different categories. We have 2 awards which are SS Parmar Research Foundation USA Prize. The first award goes to Aditya Singh from Naipur, Hyderabad. In the interest of time, I will not read the title of the paper, but I will just mention the poster number 26. The second award in this category, that is SS Parmar Research Foundation USA Prize, goes to poster 30, that is P30, Kamble Mithila Ashok. This colleague is from ISER, Calcutta. There are 2 more awards in the category, which is award category is Sati Nag Prize. The first prize goes to the poster number 37, Sushri Abhidhatri Sharma from University of Hyderabad. The second award in this category goes to poster 44. The colleague is from NIMHANS, Bangalore, Shubhadeep Datta Gupta. We also have 2 more awards in third category, which is known as SFN Bangalore Chapter Award. This award goes to poster 3 to Nikki Singh from University of Calcutta. The second award goes to poster number 14, Anam Asim from Jamia Millia Islamia, New Delhi. And the organizers have been very kind to support with 2 more awards, that is Best Paper Award from organizers. The first award goes to Jhilka Day from CSIR IICB Calcutta. This is the poster number 12. The second award goes to poster 13 to Arun Shankar from GSMC and KEM Hospital. We also have the award for the paper which is presented. Paper which was presented in the oral session. There are 2 awards in 2 different categories. The first award is Tulsabai Somani Educational Trust Award for the colleagues who are below the age of 40 years. And this award goes to Yogita Adalakha from THSTI Faridabad. The second award is in the category DM Car Prize, where the researcher should be below the age of 35 years. And this award goes to Anima Kumari from CSIR IITR Lucknow. We also have another award for the best published paper. This is known as Jyosna Mai Raghunath Bhattacharya Prize, for which I had shared the results in the GB also, but I'll finally announce the name. This award goes to Shishtha Sinha. She's from Jivaji University, Gwalior, for the paper which was published in Brain, Behavior and Immunity. I congratulate all the awardees on behalf of Indian Academy of Neurosciences, all the members, delegates, and also on my own. But see, I will also like to share the view that those who took part in the competition and due to some reason have not been able to get the award, they should not be disappointed. Believe me, the scoring was done blindly and there was no conflict at all. And I appreciate the efforts of the members of the committee, especially the Dean, Madam President, who have been very helpful to take a unanimous decision, and there was no conflict at all. Further, I would also like to share another information that we will be having the next IN meeting at ICER Calcutta. So due to COVID, as we are having the virtual meeting, I have congratulated you all virtually. Probably while we will be meeting you at ICER Calcutta, where you will have to come to receive the award. So please, I will be circulating the dates as soon as these are finalized to all the awardees. Then I will request you all to please mark your calendar in advance so that you are there to receive the award. I also take this opportunity once again to thank the organizers for hosting this wonderful webinar and appreciate these neurons which I had mentioned in the beginning round. There are two sets of, because we all have 100 billions of neurons, but the billion of neurons that Dr. Anita has and what the Dr. Shashiwala has, they are very wonderful. I must appreciate your endeavor, ma'am, because you had a very good neurochemistry and at the same time you had very good frequency also to have this conference a successful. I also take this opportunity to thank the Honorable Vice Chancellor of University of Hyderabad and the whole team which is behind as the support to Dr. Anita Jagota. Ma'am, we thank you immensely and all the members of your team immensely. And also thank Madam Shashibala Singh and their family members at NIPER who really helped us a lot to have this international webinar a successful event. With these words, I thank you all for your kind attention. Thank you.
Thank you very
Over
much.
to Dr.
Thank you very much, Professor Vinay Khanna, sir. And now it's my, and I know many students must have been enjoying now with their awards. And as Professor Vinay told, that the judges had tough time because there were close ties. And but many of you have done really very nicely and are doing very good work. But I wish if I had opportunity, the awards could have gone. I think Vinay sir would have liked to give to almost everybody. So now we request our president and co-host, Professor Shashibala Singh, to give presidential remarks.
Thank you, Professor Anita Jagota. And thank you everyone for giving me an opportunity to speak. You know, my observation is we could not, all of you, we could not serve biryani because you could not be here physically, but I think we have served you good neuroscience feast. So I'm sure all of you in last 4 days must have been very enriched and must be taking good memories with you. And while you go back, I'm sure this will be very memorable moments for you when you have listened to various people across globe. You know, the e-conference has its own merits. And some of the merits are we could listen to very distinguished speakers, not only from India, but across globe, east, south, north, and west, and which were present in the different time zones. So that was the beauty of this conference. I myself thoroughly enjoyed it. And I want to thank each one of them who could become part of this conference. A great event which was organized by my dear friend, Professor Anita Jagota. Of course, we were all advising her, and not only myself, but all like Professor Seth, Pankaj, Dr. Khanna, M.K. Thakur, we were all together with her, supporting her from each and everywhere we could. And I want to pay my thanks and also my regards to Professor P.N. Tandon, who could be part of our inaugural ceremony. It was great seeing him, and it was great seeing him on a virtual platform, and his advices, as usual, were very excellent, and I think we should sort of take them forward. We had very excellent plenary sessions by some of the very, very eminent personality from the world. We have Professor V.S. Ramachandran from University of California, well-known figure in the area of neuroscience, who has been awarded the highest civilian award from Government of India, Padma Vibhushan. We have Suresh Ratan from Denmark. We have Dr. Pavan Sinha. The way science, the bridging the gap between the basic and clinical science, he has shown practically how the basic science can nurture the clinical science and how it can really affect the society at large. The most important thing which I enjoyed was the poster sessions. For the first time, for me, it was an exposure. How do poster sessions look in e-platform? It was a really exciting journey when we were there for 2 days for the poster session, and also very well organized symposia. She has organized almost 15 symposium in 4 days. Hats off to Professor Anita. FION Symposium, then also EJN, European Journal of Neuroscience. Really great. And another thing which I want to tell everyone is, even for the Young Scientist Colloquium, we have young researchers from different parts of the world, not only from India, from Germany, from other places, from US, from MIT in US. So it was really heartening. And even now when I'm talking to you, we have almost more than 80 people joining this at this odd hours across globe. So I must be saying good morning, good evening, good afternoon. I think I would prefer to say namaskar. Namaskar, which means everything. Another thing which I want to sort of put on record is my sincere thanks to Honorable Vice Chancellor, University of Hyderabad, Dean, University of Hyderabad, and a special thanks to my dear colleague, Professor Anita Jagota. I think she may not have slept for many days. I could see her mailbox even at 2 in the night, at 4 early in the morning. And I'm sure there are 100 and more than 100 people working behind the scene. My salutation to your entire team of NIPER Hyderabad and University of Hyderabad School of Life Sciences. I'm really proud of you, and I'm sure I would like to be part of you in years to come. And with this, I want to thank Professor Seth, Senior Seth, who is the real Sutradhar of this conference. He is the real Sutradhar of this Indian Academy of Neurosciences. Sir, I can tell you from my heart, I'm speaking that it is because of you, wherever Indian Academy of Neurosciences, it is your efforts, and under your leadership, I think most of us have been working and taking it forward. I hope we continue to grow, we continue to add more members, and we continue to enrich our neuroscience community by talking good neuroscience. One last thing which I thought I will share with all of you is, since we are coming together almost once in a year or maybe sometime in a small group, can we not sort of have some kind of brainstorming session, some kind of discussion to have one flagship program where some of the groups which are working in different parts of the country can come together and have one project which can sort of address the neuroscience need of this country? Can we have a big team doing big neuroscience? And if this can be taken forward, I would like to sort of volunteer for this, and I would like to, we can have a group, we can talk about it, and we can sort of have some kind of small groups in different cities. And can we do something so that, at the end of the day, academy should grow, academy should sort of branch its trees so that the translation research finds its way to the common man. So with this, I want to thank each one of you. It has been a very learning experience, enriching experience for me personally. And also I want to thank everyone, since I will be laying off my office as President, Indian Academy of Neuroscience. I got support from each one of you, and also all of you brought different perspective for the same problem, and it really made me enriched. I also learned while working with you. Thank you so much. I also want to congratulate my new president, Indian Academy of Neuroscience, Professor Ishan Patro. I'm sure under his dynamic leadership, then the academy is going to grow, and we will all be there with you, and we will all support you in whatever possible way we can support Indian Academy of Neuroscience. Thank you. Jai Hind.
Thank you, Dr. Shashibala, for your kind remarks.
Thank you, Professor Shashibala, madam. And you can see through, you can sense through Dr. Shashibala's madam's presidential remarks that, how much energy even she has put in. And she was all the time like her blessings, her support, a single ring I will call. She may be in any meeting, but she will never miss. It never happened that she didn't pick my call any time of the day. And that was a big support. And whenever I will make a call with some stress, how to do this, she will calm me within a second. I would say that, ho jayega. Do it this way, and it will and everything went very smoothly under her guidance. So this is also another point I wanted to add, and of course I'll tell a few more remarks in the end. And with this I call to Dr. Dharmendra Kumar Khatri from Naipur, Hyderabad, to give formal vote of thanks. Dr. Dharmendra Khatri, please.
Thank you very much, ma'am. Can you hear me?
Yes.
Okay, good evening, everyone. And maybe the late evening to other part of the country, and maybe the good morning to some other part of the world. And good evening to the Honorable Chief Guest, Organizing Secretary, IAN Committee, professors, scientists, teachers, and all my dear fellow friends. First of all, I would like to thank Dr. Anita ma'am and Dr. Shashibala ma'am who gave an opportunity to speak on this auspicious occasion, and it is a matter of honor for me to extend a vote of thanks to all those who are present for the valedictory function of the International Indian Academy of Neuroscience E-Conference 2020. I, on behalf of Naipur, Hyderabad, and the entire team, the supporting team, the sponsoring organizations team, speakers, chairpersons, participants, presenters, let me call it as a fraternity of neuroscience here together, and on my own behalf, extend a very hearty vote of thanks to all speakers for gracing your important work and sharing us with your findings and opinion during 30th IAN conference. Our sincere thanks to Professor Apparao Podile, a Vice Chancellor of University of Hyderabad, for his support throughout the conference. I would also like to thank the guest of honor, Dr. M. Gauri Devi ji Madam, and Chief Guest Dr. K.K. Deepak for their valuable time. I would also like to thank Professor S. Dayananda, a Dean, School of Life Sciences, University of Hyderabad, for his support. Ladies and gentlemen, an event like this cannot happen overnight. It is just because of two young lady scientists, none other than Dr. Anita Jagota and Dr. Shashi Bala Singh, who have worked hard a day and night to make this event successful. The wheel started rolling a year back soon after the last IAN held in Delhi, and we had a personal discussion that now the opportunity on our shoulder and we have to work together. It requires a planning and a bird eye for the details that both the dynamic leader has. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues of the School of Life Sciences, University of Hyderabad and NIPER Hyderabad. I would also like to extend my gratitude to all the volunteers, students, infrastructure, and the tech support team of the University of Hyderabad who work hard in setting this up for us for the arrangement of live streaming. I would also like to thank the sponsor, University of Hyderabad, Institute of Eminence, NIPER Hyderabad, Indian Academy of Neuroscience, and European Journal of Neuroscience for their financial support. At the end, I thank one and all. Jai Hind.
Thank you very much, Dr. Dharmendra Khatri, for your vote of thanks. And as you were giving the vote of thanks, I was remembering how Dr. Shashi Bala Madam, you and me, we had visited all the halls in the university, and I was imagining here it is going to be our inauguration, and these will be the parallel halls. I had shown you our seminar hall and auditorium I showed for different events, but suddenly from mid-March, things came to standstill, and then Professor Vinay started calling and telling, no, let us try doing webinar. I was little hesitant at the beginning, but then Dr. Shashi Bala Madam said, Nahi, let's do it. So once she said, let's do it, I also said, yeah, let's do it. So first we thought it will be just two seminars a day. And by the time our EC meeting was held, which was almost last week of the June, Professor P.K. Seth sir told, no, when we are doing, let's do it in a proper way. Let it be, we should not miss a year, and let it be a proper annual meeting. So what if there is no food or fun part? Let us have an academic feast this time. And so we accepted, and under the leadership of Dr. Shashi Bala and the support from Professor P.K. Seth and Professor Pankaj Seth, Professor Vinay Khanna, and many others whom I'm Professor M.K. Thakur, and means I will call anytime, Professor Kamlesh Gulia, like all these people, I was ringing them. Sometime I took the liberty, keeping my friendship over the years, to message them at 11:30, 12, and I was surprised. They also messaged me back at that hour. So, I am really thankful to all of them, and especially Professor Vinay, who would almost every day take a feedback from me. So, Anita, what is the update? Any difficulty? He will also help pitch in and say, no, let's do it this way. Ho jayega. It will be done. And don't worry, we are all with you. So, and like I was very worried in the beginning because of lockdown, our students were not allowed. In university, we are following strict lockdown. So I was really worried, and could get my few students just 3-4 days back, and who helped me along with, of course, they were helping me from where they are, but they had electricity and connectivity issues from the remote areas, and but they helped me a lot, and I will fail in my duty if I do not thank our IT team director, Dr. Sanjay Sharma, who was also with me day and night, and day before there was little panic that there may be electricity shut down, and then for the some repair purposes he alerted me. Then I approached VC office and all, and within minutes VC solved the problem and postponed all the repairs. So I really owe a lot of thanks to University of Hyderabad, the team NIPER, who always pitched in whenever I made a single call. Shashi madam had told me, even if I am not available, for if I am in some meeting and unable to pick up call, though she always picked the call, she gave me few names, you can ring them and they will take care. But always I was lucky that Shashi ma'am personally picked up the call, and others, they, whenever I called them, they helped me and always said yes. Nobody said no, 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 or nothing like that. So I'm really, really thankful to everyone. Pankaj Seth sir also, I think I never communicated so much, but I took the liberty to message him at 11:45, 11:30, and even 1 o'clock I got a mail and message, a reply message. So I think this is very important, and this is another important thing, that through this meeting, though we got very excellent lectures, because we did not have to worry about travels or these things, but all these relations they build during our physical meetings only. And in this particular year, we enjoyed that relations which we had built, that the people promptly from different parts of the world, they had accepted our invitation. They got up at 4 a.m. in their time zone or 3 a.m. in their time zone to deliver a talk, or slept at 12 p.m. Night in their time zone to attend to our meeting. So I am thankful to almost everybody in the world who have made this happen. And I think I'm having so intense feeling that I may go on and on thanking everybody, but as good things have to come to an end. So I think I thank you one and all. And if I missed any name, it's unintentional, because the list is long, because all were my friends and all accepted the invitation readily. Professor Gurcharan, she also picked the call whenever I had any clarification to be done. So almost everyone has helped me. So with this I close, and I would say Jai Hind and Jai Bharat, and let the Indian flag fly high always. Thank you.
Namaskar. Namaskar.